This video is not on.
Communication. Are you getting anything from her? Almost.
It's him.
Audio. 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 Are you getting anything from her?
It's him now. So,
are you hearing them? Can you put them on? They're not showing just log in to say put it on.
Connected.
Jitesh?
Yeah. Hello, Mark?
Yeah, let me cut my microphone because there's a...
Sure. Okay, thanks. Keep muting.
Good. Okay. All right. So here we go. File.
Now, everybody can see you.
All right, I'll behave. Swap presenter. So let me go back to Zoom. Okay, share screen. This one, share. So is that okay? Page up.
I can see you, Mark, from Germany.
Great. Okay. Is that Astrid or?
I'm Nilima.
Okay. So, okay. So good. All right. So I'm connected, and I. What happens next? Is somebody going to introduce me, or?
I'm not sure.
Yes, yes, I'll do the introduction. Good morning, ladies and gentlemen. My name is Tepon Zwani. I'm from from NEXA. I'm an instrument scientist there. So I'll be chairing this second session of today's proceedings. Basically, we'll be continuing with the beamline instruments that were presented in the first session. We'll just be going more deeper into that. So there will be two presentations on this session. One, it will be giving an overview of the neutron instrument, scattering instrument, and their application. And the second one will go more into details where we'll see how inelastic scattering is used in study of catalysis. So the first speaker for this session is Dr. Mark Johnson. He will be joining us via Zoom. He's at the ILL Institut Langevin in Grenoble, France. Dr. Johnson has joined the ILL in first as an instrument scientist in 1995, and he proceeded to head the scientific computing in 1999. And in 2016, he was the associate director and head of science division. So currently, during that period, he played a leading role in the endurance in communication, and he's working towards increasing scientific associate of the ILL facility. So Dr. Johnson's talk will be about neutron techniques and their applications. Basically, he will give an overview of the instrument and their applications ranging from soft matter to biology and materials for energy. Yeah, over to you, Dr. Johnson.
Okay, thank you very much for that very kind introduction, and it's a great pleasure to be with you this morning. I'd love to be with you in South Africa, and I'm sure there'll be an opportunity in the not too distant future for us to meet in person. And I should apologize. I should be used to these time differences, certainly between the UK and France. And for some reason, I thought the time difference was in the other direction. But anyway, we're all together now. And I've got about 40 slides to cover neutron techniques and applications. And I'd like to take you on a journey from the neutron itself, which may be a little bit of repetition about what you've just heard and what you know, but to take you from the neutron through fundamental research and applications with industry to touch at the end on socioeconomic impact. So why neutrons? Well, neutrons are a unique probe of matter, and this stems from the unique properties of the neutron. So it has no charge, which means it interacts primarily with nuclei, and this gives a strikingly different contrast to electromagnetic radiation like X-rays that interact with the electron clouds of atoms. And what you see on the right on this slide is the scattering length or the scattering power of atoms as you go through the periodic table. And what you see is there's no specific trend. It indeed appears to be rather random, whereas for X-rays, for example, the scattering power increases monotonically as you go from light atoms to heavy atoms. So this means for neutrons that, contrary to X-rays, neutrons are very sensitive to light atoms like hydrogen, lithium, oxygen. And these are very important atoms in organic, biological material and also energy applications. But you also have very pronounced differences, for example, in these metals here around iron, which mean that these atoms can be distinguished very easily in neutron experiments, which wouldn't be the case in, for example, X-ray experiments. Because we scatter from nuclei, then obviously isotopes scatter differently, and one we're very familiar with is hydrogen and deuterium, and I'll come back to that. But isotopes also absorb neutrons differently, and there's a very useful difference there to exploit in lithium between lithium 6 and 7. And this is when we want to, for example, do imaging of batteries. And I've got an example of that to show you later. So hydrogen actually has a negative scattering length, whereas deuterium has a positive scattering length. And this means you can take, for example, a particular mixture of H2O and D2O, which will be invisible to neutrons, completely transparent to neutrons. And if in an aqueous solution you have a solute, you can actually match that water composition to one of the solutes, making that also invisible, and therefore highlighting the remaining solute. So contrast matching, as it's called, is an extremely powerful way of selecting out components in organic solutions. And finally, the neutron has a nuclear spin, which confers on it a magnetic moment. And this enables neutrons to probe directly the magnetic fields in materials. And these are magnetic fields that arise from unpaired electrons in those materials. So in this case, the neutron is interacting with electrons. So a word about neutron sources. We see here the evolution in effective thermal neutron flux, so neutron beam intensity over the years, starting with the first neutron source discovered by Chadwick. We see the evolution of reactor sources, steady state sources, with ILL offering the highest neutron fluxes, and then more recently pulsed neutron sources based on so-called spallation. And these give a higher peak flux than reactors, but the time averaged flux is somewhat lower. And the source that we're all waiting to see in Europe, at least, is the European spallation source. And actually, it should start now a bit off to the right of this diagram, the user program at the start of 2027, I think. And what's interesting about the ESS is that it will be the first spallation source when operating at full specification in the mid-2030s. It will be the first spallation source to offer a time-integrated flux comparable to that of the best reactor sources. So there's two take-home messages here. Neutron sources are large-scale facilities. That's the only way we can have intense neutron beams. And yet the intensity of those beams is only comparable to lab-based X-ray sources, good lab-based X-ray sources from today. So neutron scattering is a flux-limited technique, but that is offset, as I hope you'll understand by the end of this presentation, by the unique properties of the neutron and the opportunities that provides for research. So we produce different kinds of neutrons in terms of their energies, or equivalently in temperature, wavelength, and velocity. Reactors and spallation sources produce very high mega electron volt energies. And in a reactor, these have to be moderated to what we call to produce thermal neutrons. And this is done by the water surrounding the core. And that enables the chain reaction to propagate. But actually, we want these slower, longer wavelength neutrons to probe the materials we're interested in. And mainly we're doing this with thermal neutrons, which have wavelengths ranging from covering the range from 1 to say 4 angstroms, and cold neutrons. And there's a growing trend towards cold neutrons, and these have wavelengths from 4 angstroms up to 3 nanometers here. And for some particle physics experiments, we actually slow the neutrons down even more. We can slow them down to walking pace and do some rather spectacular particle physics experiments with those neutrons. And if I have time, I'll show you an example. So because these neutrons are only available at large-scale facilities, and they're actually quite expensive to operate, then the best way to make the full use of those sources is to surround them by as many instruments as possible. And at large facilities like ISIS, which is shown here, there are 30 or more instruments around the source or sources. So spallation sources are based on proton accelerators, and in this image, it's the same proton accelerator that's feeding these 2 sources. And on the left, target station 1 was the first source, and the focus there was more on thermal and so-called hot neutrons, and the instruments that would make best use of those particular neutrons. And then over the last decade, as I say, there's a growing trend towards using cold neutrons to look at larger scale objects and slower processes. And this target station was developed over the last decade to optimize instruments and experiments with cold neutrons. And there's a similar plot or graphic for reactors. They tend to look like this. FRM2 in Munich would look similar. And here we see there is only one source of neutrons. It is the reactor core. And emanating from there, here at ILL, we have 3 hot neutron beams, and then approximately an equal split between thermal neutron beams shown in green and cold neutron beams shown in blue. And these provide neutrons to about 40 instruments at the ILL. So a few comments on these instruments. About 2/3 of them are for structure determination, so the diffractometers, small angle scattering instruments, reflectometers, and imaging instruments. And that leaves about 1/3 of the instruments that are dedicated to spectroscopy. Elastic scattering, which is what we use for structure determination, is about 100 times more intense than the inelastic or quasi-elastic signals that we use to extract information on dynamics. And that means that the structure determination measurements tend to be much quicker, and they can use much smaller samples, which is a very important point for those people who are scientists who are preparing the samples. And another consequence of that is that powder diffraction and SANS are the highest throughput and most productive types of instrument at neutron sources. And as I've already said, there is a clear trend towards cold neutrons. This has always been a strength at the ILL. The second target station at ISIS is a very good example of that. And there's a similar project underway at SNS to have a second target station focusing on cold neutrons. And ESS will have a clear focus on cold neutrons too. So I was asked to talk about this diagram, and this is a very good point to show it, because I've shown you that the neutron sources are surrounded by large numbers of instruments. And we need many instruments to cover, to fully exploit the possibilities of research with neutrons and cover the many decades in this space and time diagram, or alternatively a wave vector energy diagram. So if we focus initially on the structure determination techniques, we see that neutrons can cover multiple length scales. And so if you look at this diagram, which I've reduced in size here on the right, if you take into account particle physics, then neutron techniques easily cover 15 orders of magnitude, which is remarkable. So this starts with nuclear structure on the femtometer length scale, wide angle diffraction on the angstrom, sub-angstrom length scale, small angle diffraction and reflectometry, which takes us through from nanometers up to microns. And then we switch into real space techniques rather than reciprocal space techniques and use neutron imaging. And there, the best resolution we can currently achieve is several microns. And of course, we can therefore go from microns up to centimeters and even bigger objects than that. So we can also do spatially resolved measurements by collimating the beam and the scattered neutron beam that we're going to detect. This is what we do in strain scanning, for example, where we do spatially resolved diffraction. And there we're using typically a square millimeter sized beam to explore a gauge volume of the order of a cubic millimeter. But we also do this in, for example, in small angle neutron scattering when we're looking at microfluidic devices. And these are small channels that allow us to progressively mix liquids, for example. And time-resolved measurements are also possible. These can go down to the millisecond time scale. And this is particularly the case if these time-dependent processes can be repeated, and therefore the statistics can be accumulated over several cycles. On the timescales, when we're interested in the dynamics of materials, the fastest dynamics we look at are sort of typically vibrations and phonons of materials. So these start at the femtosecond timescale. You might think of this as neutron Raman spectroscopy. In the next talk, I think we'll cover this very well by Dave Lennon. In many processes and materials, we're interested in diffusion. For example, it's the case of energy applications and ionic diffusion, lithium, protons, oxygen. And there the technique is quasi-elastic neutron scattering. And we can cover timescales from picoseconds out to microseconds using a range of techniques. So time of flight spectroscopy, backscattering, and spin echo, which go from the shortest times to the longest times. And one of the particularities of neutrons, and it's shown in this diagram here by the fact that these different techniques spread out not only over the time scale, but also over the length scales, it means that we're getting simultaneous spatial information from these measurements to complement the time or energy information. And so this tells us, for example, about the geometry of diffusive motions, and this is extremely useful. And there is a strong synergy, as you'll see with the examples that I'll show you, with atomistic simulations. So what we're doing in these experiments is we're measuring the positions of atoms as a function of time, but actually with neutrons, we're measuring the positions of nuclei as a function of time. And that is exactly what you calculate in classical molecular dynamics simulations. So I'd like to move on now to some applications, and this is a graphic that we put together at ILL, but it's I think it's true of all neutron scattering centers. And what this graphic tries to show is the way the facilities that we have that are shown in the middle the services that we have in the facilities, and these are shown in the middle. So we have a neutron expertise, which is in these different areas, technical areas, so nuclear and particle physics, large-scale structures, spectroscopy and diffraction, but also technical expertise in important areas like neutron optics, in particular guides that transport neutrons to the instruments, and detectors so that we detect as many neutrons as possible. But then a whole swathe of supporting services are absolutely necessary to ensure that we can deliver the best science in the most efficient way. And these cover things like sample environment, specific support for soft matter and biology, theory, industry liaison, which I'll mention briefly towards the end, instrument control, of course, and scientific computing that I was responsible for. So all of these services allow us to cover a very wide range of science. And the majority of use of neutrons covers three main areas. So it would be magnetism, because the neutron is such a unique, direct and simple probe of magnetic systems. And these cover areas like superconductors, spintronics, molecular magnets, and quantum computers. Another third of the activity covers chemistry and materials. So the next talk will be on catalysis. There'll also be a talk on energy materials, and these include batteries, fuel cells, photovoltaics, thermoelectrics, and so on. And then the third, if you like, is biology and soft matter. So there's some examples here. Host pathogen interactions is one I'll show you some examples of, because that is COVID that we're all very familiar with, unfortunately, over the last couple of years. And COVID is an example of a pathogen attacking host cells, which are our cells, and making us ill. We also have in biology radioisotopes for therapeutic applications and boron neutron capture therapy. And this is actually work that comes from at a place like the ILL, our nuclear and particle physics group. So this is a smaller activity at the ILL, but nevertheless a very important activity. And there's some very exciting science which is probed with, which uses the neutron as a probe in particle physics. And I hope to show you one example of that. So I'd like actually now to focus on battery research, just to take you through the range of experimental techniques. I wondered how I could best do this, and rather than just showing what I thought might seem to be a random selection of examples to illustrate science and techniques, I thought I'd try and have a theme. So I'll take you through batteries, through some structural work, and then some looking at dynamics that's relevant to batteries, and then show you a few similar examples in the same way on health and soft matter, and that focuses on COVID, because that's extremely pertinent at the current time. And then hopefully I've got time for a couple of examples on magnetism, but Martin Böhm will show you a lot more of that tomorrow. And as I said, one example on nuclear and particle physics. So batteries, battery research really needs a multitude of techniques, not just neutrons, but within neutrons, we need a whole range of techniques. And that's because there are really a number of points of interest. First of all, there are many length scales, and these are sort of illustrated in this cartoon. You know, we have the electrodes shown as hexagons here. Between them, there's a separator, but there's also the electrolyte. There are important changes in the electrodes, structural changes, but also volume changes, because lithium is going to flow, for example, in lithium batteries, is going to flow in and out of these electrodes. There are important effects around the interfaces between the electrode and the electrolytes. And then, of course, diffusion, which we'll measure by quasi-elastic neutron scattering, is crucial to the flow of electrodes through the electrochemical cell. So, starting at the shortest length scale. So this is some diffraction work looking at the microscopic structure of a manganese oxide electrode material as lithium flows in and out on charging or discharging. And first of all, if you want to do this operando work, well, you need an electrochemical cell which is adapted to the neutron beam and instrument. And that's shown here on the left. There's some background noise. I don't think somebody wants to ask a question yet, but maybe there's a microphone on that could be cut. So this electrochemical cell, there's a lot of components to it, but the key part is this sort of pellet in the middle here, which is shown here as a photograph. It has the electrodes, separator, and electrolyte. And the point here is that in order to work, it has to be very thin, less than, well, typically about 1 millimeter. And this means we're only going to subtend a very small part of the neutron beam. So that makes the experiments rather challenging. But nevertheless, there was some beautiful results obtained from this work, where we see how this lithium manganese oxide electrode material undergoes important structural changes, two structural changes, as the lithium is completely removed from the electrode. So here we see one Bragg peak and its discontinuous evolution, which shows clearly these two structural changes. And what's very interesting is that if you change the composition of this electrode material, you start to add a bit more lithium and remove some of the magnesium, manganese to compensate. Well, you can't quite remove all the lithium, but you can actually remove one of the phase transitions. And if you go a little bit further, you can stay in the alpha phase over a much wider range of lithium composition. And that's shown here by this smooth evolution of the Bragg peak. And you might wonder why this is interesting. Well, one of the key factors that's of relevance to batteries is their cyclability. And their cyclability is improved if these important mechanical changes are as smooth as possible on cycling, on charging and discharging. And that's clearly the case here with this lithium-rich composition of the electrode. So there's considerable interest in silicon as anodes, and they can be in the form of nanoparticles. They would have higher specific capacity, but actually, as we'll see in the next example, one of their problems is cyclability. But for these nanoparticle anodes, well, the technique you then want to use is small angle neutron scattering. And this is the cell that was actually used, and it's turned perpendicular with respect to the cell that we saw on the previous slide. And that's because in small angle scattering, we're going to measure small deviations of the neutron beam passing through the electrochemical cell. So that's illustrated here in part B of the figure. And you see more details of this thin electrochemical cell in part C of the figure. So the cell works from an electrochemical point of view. You can discharge the cell, which is which corresponds to lithiation of these silicon nanoparticles, and then you can charge or delithiate the nanoparticles. And there is a corresponding systematic evolution of the small angle scattering signal. This shoulder grows in this sort of intermediate Q range, and then it disappears again on delithiation. So this can be analyzed. You can fit these curves, and the model that's used to fit these curves is a core shell model. And here you see the length scales. We see that the nanoparticles are about 160 angstroms in diameter, and as they're lithiated, well, this core grows. The shell grows in thickness, and the core correspondingly decreases in diameter, and that's illustrated at the bottom. So a great deal of lithium is incorporated in the shell of these silicon nanoparticles during discharging. So that's some unpublished work, but there's been some very detailed work using reflectometry to probe the way lithium goes into solid silicon anodes. And the table here at the top right just gives an indication of the volume change, and therefore the mechanical changes that can arise in these systems. And so the one we're interested in this work is lithium entering a silicon anode. So this is reflectivity work, 2 publications by pretty much the same team. And what they did is they put together another kind of electrochemical cell optimized for reflectivity experiments, where we have an incident beam and we look at the reflected beam as a function either of the this reflection angle theta or the wavelength of the incident and reflected beams. And this gives us Q-dependent information of the reflected signal. You can't see the data. This should be animated, but behind, or you can just see some of the data in the top right here. And there is a systematic evolution of these reflectometry or reflectivity curves as the anode is lithiated. So what we see, what we can do with these reflectivity curves is transform them into scattering length densities as a function of time. So we've got here the scattering length density is shown in color. Where we have a lot of lithium, the color is red. We show the vertical axis shows the distance in nanometers, the extent to which the lithium penetrates into the silicon anode, and then the time scale associated. And so it's about 10 hours to cycle this device and to push lithium into the silicon anode and then to remove it again. So what's interesting here is, first of all, there's a sort of a dummy cycle. Then we load lithium into what's at this point silicon, crystalline silicon, but actually after removal of the lithium, well, the silicon becomes more and more amorphous. We see that these cycles, they don't repeat in exactly the same way. There's more penetration as you go further. But what's really interesting in this work, and the focus of this work, was what happens at the solid electrolyte interface, and that's shown here. And this is these are the changes that are thought to block further lithium penetration into the silicon anode on cycling, and therefore limit the cyclability. So moving on to imaging, in this case, the samples are much simpler. There, you simply take a battery of this kind and put it in the neutron beam. And you'll hear more about this tomorrow from, or later today, from Nikolai Kardylov, who is really an expert on imaging. And this is a technique where we take advantage of the fact that you can have lab-based X-ray sources that are comparable in intensity to our neutron beams. And this is one of the instruments where we combine in situ neutron and X-ray imaging. But the work shown here, the X-ray imaging, which is shown on the left here, was actually performed at the ESRF. And you see exquisite details about the way the electrode in this battery changes as the battery discharges. There is already cracking in the electrode materials due to manufacture, but this gets worse, significantly worse, during discharging of the battery. But what you don't see in the X-ray data is the other anode, which in this is the other electrode, which in this case is made of lithium. And there you see it here. It's shown in white in these corresponding plots here. But also the electrolyte and the center of this type of battery is an electrolyte reservoir. So one of the major effects that you see in these batteries is the consumption of the electrolyte going from left to right as the battery is discharged. So you see a large amount of this electrolyte has been used up. And there are similar major structural changes and deformations seen in the neutron imaging data, which complements perfectly the X-ray imaging data. So you'll hear more about that from Nikolai. A couple of slides now to finish the battery examples on dynamics. As I said earlier, one of the fundamental processes is lithium diffusion between electrodes. And I'm not aware of any work where this has actually been measured directly, but there has been some very nice work performed at ISIS on sodium ion diffusion and in sodium cobaltate. And this was also combined with ab initio molecular dynamics simulations. So what we see here on the top left is quasi-elastic neutron scattering data, and you have to look quite carefully to see the signal. It's here shown in the inset. It's in the foot of the elastic peak in the so-called quasi-elastic part of the spectrum. And it's this broadening of the elastic peak that is an indication of diffusion in this sodium cobaltate material. It's the sodium that's diffusing. So there is only a weak and limited diffusion in the low temperature ordered phase, but as you go into a so-called disordered stripe phase and then a fully disordered phase, there is a significant onset of quasi-elastic scattering. And from this, you can extract a diffusion rate or constant, which is highest in the disordered phase. This is superionic diffusion. And we have quasi one-dimensional diffusion in the disordered stripe phase and essentially no diffusion in the ordered low temperature phase. And this characterizing the geometric nature of the diffusion processes is helped by this Q dependence here of the quasi-elastic signal. So that's some very nice work supported by diffuse X-ray scattering and ab initio molecular dynamics. And finally, batteries, I'd like to show you just a piece of inelastic neutron scattering work. So this is moving to higher energies now, collaboration between Indian colleagues, colleagues in the US and also at the ILL. And the material here is lithium aluminium oxide. So lithium is shown in blue here. The aluminium oxide tetrahedra are shown in purple, and so red, the red atoms here are the oxygen atoms. And what they looked at by combining inelastic neutron scattering and ab initio molecular dynamics is the correlation between lithium diffusion and the librational and vibrational motion of these aluminium oxide tetrahedra.
Yeah, thanks a lot.
And so what we see here, we want to focus on the experimental data. So here we see the vibrational density of states of this material as a function of temperature. And we see basically some of the vibrational peaks are more strongly attenuated as a function of temperature than others. And to know which are those vibrational peaks, well, you need a calculation which enables you to separate the density of states into the so-called partial density of states. And that is shown on the right. So we have the experimental data at low temperature shown in red, and the corresponding calculated data shown as the red line. And there is reasonably good agreement, which gives you confidence in interpreting the experimental data from these ab initio molecular dynamics simulations. And it's the black curve, which is the partial density of states for lithium, centered around 40 millielectron volts. And these are these are the modes that are strongly attenuated with the onset of temperature and the onset of lithium ion diffusion. So this is a clear indication of lithium ion diffusion in these vibrational density of states measurements with neutrons. So that's a range of battery examples covering structural effects over a wide range of length scales and dynamics over different time scales. I'd like to spend 3 or 4 slides focusing on COVID. I'm sure we've all had COVID. I had it in the summer. And it's been a nuisance to us, but it's also been an opportunity to show, how important research is, including research with neutrons. So again, starting at the shortest length scales, I'd like to show you some, very briefly, some work on protein crystallography. So this was again a collaboration between SNS and ILL. And it uses Laue neutron diffraction using cold neutrons because we're looking at large scale structures, in particular proteins here. And this work focuses on what's called the main protease, and it's the enzyme that cuts up polypeptide sequences and allows the virus to replicate in our cells. And this work recently evolved into looking for drugs and actually repurposing drugs, for example, that have been used to treat hepatitis, and to see how these drugs can enter into the active site of the main protease and block its action. So a lot of information is typically gained by X-ray diffraction, in particular at synchrotrons. There's really an industry going on there, but neutrons give absolutely key insight in these drug-host interactions because they're mediated often by structural water and hydrogen bonds, and it's only neutrons that can show you where those key protons are in these processes and mechanisms. We've probably all had vaccines as well, and some of these vaccines were based on messenger RNA. Actually, not the ones by AstraZeneca, delivered by AstraZeneca, but they have been working on this type of vaccine, and they published some work in 2021. And so this is a collaboration between AstraZeneca, Sweden, ANSTO, and ILL. So again, international work. And here, the focus is on the structure of these lipid nanoparticles, and in particular in the presence of proteins that are present at the uptake site, and how those proteins interact with the lipid bilayer and other molecules in the bilayer, like cholesterol, for example, and how this therefore mediates the release of the messenger RNA and its effective action as a vaccine. This work, the bit of the work that was done at ILL was done on D22, and I just want to show this photograph again, because again, here we've been able to, in terms of instrumentation, have this very nice in situ combination of neutron and X-ray small angle scattering. And as a final example, we've heard about the spike protein. This is the way the infection of our cells is initiated. And there was 2 pieces of work done here, which were published in JACS. And these focused on the way what are so-called fusion peptides, so polypeptide sequences in the spike protein interact with lipid membranes. And these are either in the form of liposomes, so spherical nanoparticle structures. And here we see the small angle scattering data and the way different fusion peptides interact differently with these liposomes and essentially allow the virus, the pathogen, to interact and its viral load to penetrate our cells. And there was also some reflectometry work and actually quasi-elastic neutron scattering work, but it's not shown here. And the reflectometry work in particular show very nicely how this fusion peptide enters into the lipid bilayer, and this is actually mediated in some way by the presence of these calcium ions. And so this fusion peptide, it really can enter completely into these lipid bilayers. So I'm keeping an eye on the time, so I think I will leave these two magnetism examples to be covered by Martin Böhm. This one in particular it's a beautiful example of work on highly relevant ferroelectric multi-ferroic materials here, copper iron oxide. And it's a very challenging experiment done with polarized neutrons and the most sophisticated technique that we have available in our facilities, which is spherical neutron polarimetry. And one of the other remarkable things about the work was that they used a very, very small crystal, 0.04 cubic millimeters. And with that, they were able to explore this very rich phase diagram. And it's only neutrons that can give you the details of these magnetic structures in these different phases. And they vary from collinear structures to spiral and ellipsoidal structures. So a beautiful piece of work that, if you're interested, you should look at in Nature Communications. And work, another piece of work on magnetism using inelastic neutron scattering, and that's to study skyrmions. These are large-scale structures that have been studied with small-angle neutron scattering, but here it was inelastic work, and Martin was involved in this, and so maybe he will tell you more about this. And the reason for doing the inelastic work in this case is to understand the magnetic interactions that give rise to the anti-ferromagnetic ordering between these spiral skyrmion structures. And that's what's unique in these materials, and it's the point of interest in this work that was published in one of the Nature journals. So, particle physics, I don't think I'll have time to go through that. So I think I'll move on to a few comments about the examples I've just shown you. One of my predecessors as science director, he coined an expression which was more than just neutrons and the instruments I've shown you. You need a lot more than that, as I mentioned at the start, to do research with neutrons. Sample preparation is essential, especially deuteration. That's highly relevant to the COVID work that's been shown, that I've just shown. And for soft matter in general, you know, these samples are intrinsically fragile, and we need a lot of ancillary equipment to ensure, to prepare, test and characterize the samples before, during, and after experiments. Sample environment is crucial. The skyrmion work and the other magnetism work on the multiferroics was performed with high magnetic fields and high pressure. I've shown you operando work and in situ experiments combining neutrons and X-rays. So actually at large scale facilities, we have large teams that support this important activity. So at ILL, ISIS, SNS, for example, there would be 25 technicians and engineers that are supporting precisely this sample environment related activity. And finally, something that I was closely involved with was data treatment, and essentially how we transform data into science. And ILL is part of the MANTID collaboration. So this project was initiated at ISIS, taken up by SNS, and ILL is the reactor source which has fully embraced MANTID. And so this allows us to share a lot of expertise and development and provide a seamless experience as users move between the facilities. I've mentioned the importance of simulations, and what came up during COVID was the fact that all of this software, including instrument control, could be made available remotely so that experiments could never still be performed without all the users coming to the facilities. So that offers very interesting perspectives when large-scale facilities are remotely located and moving forwards. So if the chair could stop me or tell me when I've got a few minutes left, but I'd like to cover two more points, and these are neutrons and industry, and then touch on some socioeconomic impact. So neutrons and industry, we tend to think of this in terms of proprietary beam time. This is often related to research at higher technology realization levels. So this is where companies are going to buy beam time so that they can actually keep the data to themselves. And typically, large facilities are selling tens of days per year in this way. And the experimental techniques, this is data from ILL that were most used up to, I think this data covers the period up to 2019, was small angle scattering and stress scanning, so engineering diffraction. And the sectors, industrial sectors or areas that were covered were dominated by energy, engineering, of course, for strain scanning and a range of other applications. But what's happened recently is at ILL in particular, but ISIS is also... yeah.
Dr. Johnson, sorry to come in. Is it possible that you can wrap up in the next 2 minutes or so?
Yeah, I'll go quickly and I'll skip some slides. Yeah. So the new opportunity that's being explored at ILL and ISIS, but it's also been explored for a long time at PSI and Munich. You'll hear from Ralph Gillis. Is neutron imaging, and this is giving us new perspectives for significantly increased revenue. It is taking over from SANS as the most widely used technique, and the area industrial sector that we're working mainly with for neutron imaging at the moment at ILL, along with PSI, is the aeronautics and space industry. But industry can use our facilities with academic users on the basis that their data will become public and they'll publish their results. And this graphic here shows that of the 1,200 proposals per year that we receive at a facility like ILL, well, actually, in 2020, it reached a level as high as 17% of these proposals have direct or indirect industry involvement. So that's a really interesting to capture. And we'd like to know who these companies are, and we have different ways of doing that. And we did manage to establish a list for the period 2011 through to 2018 of 102 companies and research technology organizations that use ILL. And finally, neutrons are not well known to industry, and industry would much rather use would much rather use lab-based techniques like X-ray techniques. So we do need to have a long-term perspective on working with industry and actually seed people, have collaborative projects and seed people in industry. And here I want to highlight a PhD program that we set up with our neighbor facility, the ESRF, called Innovaction or InnovaXN. And we have 40 PhD projects, each one having an industry partner. And these are the industry sectors covered in that program. So mainly energy, catalysis and chemistry, pharmaceuticals and biotechnology. I was asked to mention socioeconomic impact. So there was an excellent report that was developed by STFC, the Science and Technology Facility Council, for the ISIS facility in the UK. And I actually, when I was the UK Associate Director, I had to do something like this as part of the business case for continued funding for ILL for the next decade. So the skill development, education I've just mentioned, we train PhD students, but actually we do this directly, but also indirectly, because hundreds and hundreds of our users each year are PhD students. But our facilities are highly valued, high-tech, international working environments, that provide a unique opportunity for training highly skilled staff. So this I'll go through very quickly. Industry use demonstrates short-term relevance and impact of neutrons, but it cultivates leading energy sectors. And what was done in these impact studies and business cases was to estimate the impact of neutrons with financial considerations as well. So Strain scanning I've mentioned, and this was used to look at welds in reactor materials and therefore extrapolate and indicate a possible increase in reactor lifetimes. And so this offers an enormous saving in billions of euros in terms of reactor operation, and of course in terms of climate, offers extending low carbon energy to millions of homes. So there was similar work for in the railway industry worth 50 million euros, and neutrons are responsible for a part of those savings. Spider silk. What's interesting is that some of this work is actually upstream and doesn't yet involve industry partners. So here, neutrons and research are generating knowledge which has long-term impact for societal challenges. So here there are examples on biodegradable orthopedic implants, spintronic materials like the skyrmions I mentioned, carbon dioxide sequestration, and there's a potentially huge market there, as you can imagine, and organic electronics. And since we've just had the Nobel Prizes, I wanted to finish with a couple of examples on the Nobel Prizes, because we tend to think of this as being fundamental research. But if you look at the press releases, of course, the Nobel Prize Committee, an organization, obviously wants to reach out to the general public, so they highlight the applications that are possible. So in 2016, there was a Nobel Prize awarded to these three eminent scientists, Taulis, Haldane in the middle, and Kosterlitz. And they worked on unusual electronic phases and states of matter like superconductors and thin magnetic films, topological phases. And at ILL, we were particularly proud of this because Haldane started this work as a postdoc at the ILL. So neutrons are relevant to Nobel Prizes, and there are future applications. And in this case, it's material science and electronics, or indeed spintronics. And the final example is, of course, this year's Nobel Prize in physics. And you might not think there's a connection to neutrons here, because the work that was rewarded was entanglement of photons, so the quantum properties of photons. And the applications that are expected are in quantum computers, quantum networks, and quantum communication. But one of the Nobel Prize winners was Anton Zeilinger from Austria, from Vienna. And he actually spent 15 years at ILL looking at studying the quantum properties of the neutrons. So there really is a strong link between neutrons, its research education, but also innovation, and this is in some way encapsulated in these two Nobel Prizes. I'm afraid there's been a mistake with my conclusion. I actually had a problem starting my computer before joining you and didn't have time to finish the conclusion slide. So I'd like to conclude just by saying that I hope I've given you an overview of neutron techniques and applications, showing how they can be used for fundamental research through applications with industry and generate socio-economic impact. And in terms of prestige, from time to time, this materializes itself in Nobel Prizes, which have clearly used neutrons in their work. So with that, I'd like to finish and thank you very much for your attention. Thank you.
Thank you, Dr. Johnson, for the interesting talk. I think personally I've learned a lot on the different techniques of neutron scattering and the application. Although we are pressed for time, I think I'll allow one or two pressing questions from the audience. Are there any questions for Dr. Johnson? Any questions? Okay, just one question from me. The work you've presented on battery, you've showed that you are using different techniques, multiple techniques. How many of those techniques were from the same instrument? And what are the disadvantages of having one instrument being able to be used for different techniques, accommodate different techniques?
That's a very good question, because I think I showed you 5 or 6 examples of battery research covering structure and dynamics. And actually, each example was using a different kind of instrument. Ideally, you'd be able to use one instrument to get all of that information, but it's not possible. And generally, for all research with neutrons, when you want to cover different length and time scales, you need to use several instruments. It's a particularly poignant remark in that there's a European project at the moment called BigMap, as part of the Battery 2030 Plus roadmap, which has been launched by the European Commission. And the work we're involved in there is to try and design electrochemical cells that you can use on more than one instrument. Because what you'd like to do is take the same sample, 2 different instruments, and when your sample is a simple powder, that's possible. But when you have an electrochemical cell and the geometry of the instrument's very different for a powder diffractometer and a SANS instrument and a reflectometer, then you actually need, at the moment, we're using different cells for each instrument. But when you want to look at, for example, battery cycling over long periods of time, and on different instruments, in order to ensure reproducibility of experiments, we'd like to do it with the same cell on different instruments. So we can't use one instrument, but we would at least like to use the same electrochemical cells, not only for the different neutron techniques, but also to go to synchrotrons as well, for example. So that's one of the key points, is being able to study exactly the same system on a range of different instruments.
Okay, thank you very much. Any question? Yes.
Thank you so much. Thank you so much for the presentation. I wanted to know, do you have any examples for the geosciences to use in geosciences?
There certainly are examples. And you may hear from Nikolai Kardelov in imaging, because we have developed our imaging activity recently, as I said. And we started that initially with a team from the local university, and their work is on geosciences. So, you know, their core research work is in geoscience, and so they're looking at, for example, the penetration of water into rocks, and that's related to fracking. Related to that, of course, there's lots of work on concrete and building materials, which is probably not geoscience, but it is, if you like, rocks and hard materials, and the presence of water in concrete, which can have spectacular effects. For example, the collapse of the Twin Towers was thought to be due to the heating and vaporization of water in concrete. So there's 2 examples. There's also work in mineralogy, looking at the the atomistic microscopic structure of minerals with neutrons. You may call work with clays geoscience. Clays are often used, for example, for water purification, and so there's. I'm aware of work a bit related to what I think Dave Lennon's going to show, where we use vibrational spectroscopy to characterize the way molecules are trapped in porous media, and clays are a good example of porous or confined media. So there's clays, minerals, water in rocks, and concrete as sort of different examples of geoscience that I'm familiar with neutrons.
Thank you so much.
Okay, I think in the interest of time, I'll stop here. If there are any question in the chat box, my colleague will forward them to you, and you can reply on them. So we are way behind on schedule. So I'm just going to go ahead and introduce the next presenter. That is Professor David Lennon. He's from the School of Chemistry at University of Glasgow in the UK. He works mainly on surface chemistry and looking at different spectroscopic techniques to study the interaction between the atoms and molecules on the metal substrate. So he will present a talk on the use of neutron scattering techniques in catalysis, focusing on two examples: Fischer-Tropsch ion-based catalyst and the methanol to hydrocarbon Zeolite catalyst. Are you there, Professor Lennon? It's not. Okay, maybe in the meantime, while we are still waiting for Professor Lennon, I can just mention that there will be a photo session after this last talk before lunch. I understand we will stand in front of the building over there, but I think the organizers can also clarify that where we are going to have the photo session. And after this presentation, after the photo session, we will break for lunch. And I'm just going to request that in the interest of time, maybe we can reduce lunch so that we can get back on schedule.
I don't want to take up more time since I didn't join very efficiently the meeting. But there's a question in the chat. So while we're waiting for Dave Lennon, maybe I could I answer the question in the chat?
Yeah, I think you can go ahead.
Okay, so it's from Daniel Wamwangi. So you ask about the sample size dependence with the different neutron techniques and the flux of neutrons required. So as I mentioned at the start, if you're doing elastic scattering or structure determination, then that signal, as I say, it's 100 times stronger than the quasi-elastic or inelastic signal that you would use to study dynamics. So it's in those structure measurements that you can use the smallest samples. And so the crystal for the multiferroic diffraction work was 0.04 cubic millimeters. The crystals used for protein crystallography, so there it was the SARS-CoV main protease, that's about 50 to 100 micrograms, I think. So it would be a very big crystal for X-rays, but for neutrons, it's a tiny crystal. And in all single crystal work with neutrons, it is these days a challenge to grow the crystals because the X-ray techniques have developed so strongly that the expertise in crystal growth is diminished. The flux required, the flux at the ILL, if you remember the plot I showed, it's 10 to the 15 neutrons per square centimeter per second in the reactor. But the best neutron flux we get to an instrument is about 10 to the 10 neutrons per square centimeter per second. And that's what we have on the imaging beamline. And the flux is high on imaging beamline because there's no pulsing of the beam or no wavelength selection. So we use absolutely all the neutrons that are coming from down the guide. And these guides now have been, carefully designed, and they're very high performance guides these days that really transport a lot of neutrons. And then as you pulse beams and make wavelength selection and collimate beams, where you of course you reduce neutron beam intensity further, and that typically goes down to 10 to the 6 or 10 to the 7 neutrons per square centimeter per second. But one key point that I'd make is that at the ILL, we're very fortunate to have this fantastic neutron source, but the neutron source has never increased in performance over the years. That's not possible. But what has changed dramatically is the transport of the neutrons from the reactor to the instruments, and of course the performance of the instruments. But it's so, you're talking in South Africa about having a new reactor which would have lower flux, but by optimizing these guides, as has been done at all neutron facilities over the last decade or so, you can have incredibly efficient instruments at much smaller facilities than ILL or ISIS or Munich. So I think there's really fantastic opportunities for you there to design an instrument suite and have really world-class instruments at a smaller research reactor. So I see Dave's arrived now, so I'll mute and stop the video.
Dr. Johnson, if I can just come in, the next speaker is online. Can we take this opportunity to thank the previous speaker again?
Thank you very much. It's been a pleasure. And if there are any other questions, please send me an email or we can do it via chat while the session's live. And thank you again.
Thank you. Thank you. Prof. Lennon, are you online? Prof. Lennon?
I apologize. I'm sorry. I've got my times mixed up.
Yeah.
I'm really sorry.
I've already done the introduction. I think you can just proceed if you are ready.
Okay. Can I? Thank you. Can I share screen? Yeah.
Yes.
Okay. I'll just open it up. Sorry. I'm just a little bit. I've just come in from a lecture. I've got my times wrong. I'm really sorry about this. It's all. I've got my talk ready.
Yeah. So your talk is supposed to be for 30 minutes plus 10 minutes for questions.
Yeah. And how long would you like me to speak for?
If you can stick to the times, that would be great, because we don't want to move away too far from the program.
So I'm totally confused with time. Can you tell us how long would you like me to speak for, please?
Sorry, can you repeat the question?
Yeah, how long would you like me to speak for?
How long?
Can I speak for 30 minutes or 25 minutes or?
Say 30, 35.
Okay, that would be wonderful. Okay, thank you. And I must apologize to everybody. I'm sorry, I've just come back from a lecture and I've got my times wrong. Sorry,
yes,
is that okay? Can you see my picture and can you hear me okay?
Yes, we can see it.
Okay, look, thank you very much. And I'm sorry I missed Mark's talk. That was a shame. Right, okay. So apologies to the audience and apologies for my chaos. It's me trying to run teaching courses at the same time. What I'm going to do, I'm going to go through to try and show you an illustration of how we can use neutrons and link it in with economics of society, in particular with regards to chemical manufacturing. If I overstay my welcome, please stop me and I can stop at any stage. What I'm going to try and do, I've skewed it a little bit towards South Africa, and then I'm just going to create a little bit of diversity at the back end to show a different sequence. So some of you might recognize the picture in the front. On the front, this is actually from Sasolburg. I believe it's from Sasolburg. And what I'm going to show you is how neutrons have helped understand some of this contributor to the understanding of the chemistry associated with contributor to the South African economy. So I'm going to look at the application of neutron scattering techniques in heterogeneous catalysis research, and it's a pleasure to be here. So what I'm just going to refer to now, I've got two subsections, and the first one is, I'm sure everybody in the audience is familiar with Fischer-Tropsch synthesis. And what I thought I'd do, I'd show you how neutrons can relate to that aspects of even quite mature and developed processes. And what you're seeing is there's a real future springboard here, is I think that there's this opportunity for where does hydrogen sit in our chemicals landscape, in our energy, in our fuels. And so the illustration here, I'm sure, no, I don't need to, this is coals to Newcastle. Here we can take any source of carbon. We need to gasify it to produce syngas. Syngas is carbon monoxide and dihydrogen. And then we can put it through this amazing process, which is called Fischer-Tropsch synthesis, which Sasol in South Africa are experts in. And we can produce a whole range of materials. In fact, the products that come out of this is what's called a product slate. And you can make chemicals in olefins, you can make gasoline, you can make wax and lubricants, and there's a whole range of materials. And so this is a very mature process. And this was a collaboration between the EPSRC, the ICIS facility in the United Kingdom, and Sasol Technology, who had a branch in the United Kingdom. And these are the people that are involved, and they're all sterling good folk. Okay, so just to set the background, is that some time ago we did some fundamental studies using neutron funded by the EPSRC, and we were able to show that we could look at deactivated catalysts. And these are industrial grade catalysts. And on that basis, Sasol said to us, well, could you look at one of our Fischer-Tropsch catalysts, that in fact this catalyst, they gave us a sample from their Secunda coal to liquids plant in South Africa. This is their advanced synthol catalyst. And could we see if we could obtain any information via the neutrons? And so what we have here is that the sample was really black, it's very dark, and we didn't really expect to see anything. If you try the infrared, you can't see anything. However, the neutrons can reveal the vibrational fingerprint of the actual catalyst post-reaction. And this one has just been extracted in toluene, and this is dichloromethane. This is to take away the main products of the Fischer-Tropsch process. And what are we seeing is the residual material of the functioning catalyst. Now, this was really quite a surprise to us, and this is quite a rich spectrum. And underneath here, just at the bottom, it's all been written up in publication. The table here is showing you how we can assign nearly all of the bands. And if you accept that Mark will recognize this, that our Tosca spectrometer runs out of resolution a little bit above 2000, we can see that we've got not great resolution in the CH stretching region, but we really have quite good resolution in the deformation region. And we can assign a whole series of bands. And the spectrum, we took some time to do it. We had to do some modeling, but really we felt as though that we could describe what was the working format of this catalyst that had been making as part of an active Fischer-Tropsch synthesis. So interesting, the spectrum cannot be assigned to a single molecular species, but it indicates the presence of what we call a hydrocarbonaceous overlayer. And in the heterogeneous catalysis groups, say in the United Kingdom, we're familiar with this type of signature, and we recognize what it means, and we can understand how it might perform a function. And so overall, we would say that the spectrum is consistent with partially saturated polycyclic hydrocarbons. And then we can speak for our industrial partners and say that how we might relate that to the catalyst lifetime. So this talk is primarily going to be based on iron-based Fischer-Tropsch catalysts. And just to let you know that we've picked a generic reaction scheme. So if we did full-on FTS synthesis, we would just make complex hydrocarbons, and we would swamp our INS spectrum. So what we do, we feature this spectrum, this reaction here. It's actually just a CO hydrogenation reaction. And essentially the product is just methane. And what it's done, it was originally set up by some workers in Spain as being indicative of iron and syngas chemistry. Okay, so that's what we're going to use. And excuse me, the benefit of this is that the product doesn't get in the way of the spectrum and of the changing of the evolution of the catalyst. So just I'm not going to show you any INS spectra straight away. I'm going to try and show you how the catalyst changes during the course of it being. It's actually the catalyst that's put into the reactor is a pre-catalyst, and what it does, it actually changes into the working catalyst, and it does this evolution. And what we have on the right hand side here is temperature programmed oxidation as a function of time on stream in the reactor. And what you're seeing, so this is actually revealing the extent of the change of the carbon on the active iron Fischer-Tropsch catalyst. And what we see, this is 3 hours, 6 hours, 12 hours, and 24 hours. And I hope you can see that there's dramatic changes in the carbon over this period of time. Just for instance, the reaction takes about 6 hours or so to reach steady state, maybe a bit longer, but you can see the catalyst is changing all the time. And what we've identified is alpha, beta, and gamma peaks. And the alpha is what's a reactive species. You'll see that goes after a period of time. There's then it forms amorphous carbon, which is this beta species. And then underneath it starts to form iron carbides, and these constitute to some extent the active phase. So we're seeing considerable change in the catalyst as a function of time on stream. And so we could follow the carbon, but what's the situation with regards to hydrogen? And this is why we use neutrons. And just the hydrogen is actually very difficult to measure in an industrial process, and what tools would we use? And it's also because if you produce water, it's very difficult to do a hydrogen mass balance. And one of the benefits of the neutron technique is that we can actually identify how hydrogen is dispersed between the catalyst matrix post-reaction. And this is information that is very difficult to get otherwise. Now, what this is recorded, I'm not sure, oh, yes, it's recorded on the MAP spectrometer, and this is the high energy scan, and you can clearly see the CH stretching region, and we can distinguish two bands. 2934 is associated with sp3 hybridized carbons, that's the CH stretch for that, and the band at 3052, which is dominated, is due to sp2 hybridized carbon. So that's due to alkene or aromatic species. And so what we can do, we can see the growth of that as a function of time on stream, 3, 6, 12, and 24 hours. And that really is quite a strong spectra. Okay, so we see increasing quantities of aliphatic of these species. And how can we signify that? One of the benefits of the spectrometers that we have at ICIS, we can switch across to, we can change the incident energy, the incident neutron energy. And what we do is we maintain resolution over a different region of the spectrum. There's not many spectrometers in the world that can do this. And this allows us to gain really, really important chemical information. And what we can see here, we can see some deformation due to aromatic species. We can actually see a phonon mode. This is at 591 of the magnetite. We originally starts with hematite, so we can see the transition across to of the material that's associated with the iron. And then there's a number of species which are CH deformation bands that we can associate with alkenic species, and also an associated magnon associated with the magnetite. Okay, so you can see that we're starting to get really rich information on these spectra. Now, one of the take-home messages that we must recognize for inelastic neutron scattering, that if we do it properly, it can be quantitative. So not only do we get a qualitative image of what's going on the surface, we can actually be more precise about that. And in this case, excuse me, this is the 24-hour spectrum. We've curve fitted it, and we've curve fitted it to the two bands, to the saturated CH stretch and the unsaturated CH stretch, saturated CH stretch, unsaturated. And you can see quite readily, we can mimic the surface, we can fit them quite readily, and we can quite readily curve fit them. And then we have to do something that takes a little bit of time, it takes a bit of effort, but this was developed a few years ago as part of a separate project where we were able to quantify and calibrate the MAP spectrometer, the amount of hydrogen that we observe in the spectrum. And here we're using polystyrene as a calibrant, as an internal reference. And here is the spectrum for different masses of polystyrene. You can see we can actually record spectra down at really quite low levels. And what it does, it allows us to determine a sort of a response factor for the hydrogen in the spectrometer operating under these particular conditions. So this is a calibration curve for the concentration of hydrogen. And this means then we can convert our INS calibration to the extent of retained hydrogen. And believe you me, for engineering senses, this is very important. And so here in the left-hand side is our previous set of INS spectra recorded at the high impact energy. And so this corresponds to the CH stretching region. We can resolve out the unsaturated from the saturated. And here we see the sp3 band, and we can see that over a period of time, this saturates, and that's rather interesting. But if we look at the sp2 hybridized CH stretching region, over the full 24-hour period, it looks like it's slowly starting to saturate, but you'll notice that it's clearly got a different profile to that for the saturated species. And so we're making the conclusion that the aliphatic component saturates at around about 5 hours time on stream, whereas the aromatic olefinic component of the hydrocarbonaceous overlayer continues to develop around about 24 hours and probably a little bit further than that. And this information wasn't previously available before, and it allowed us to sort of surmise how the catalyst might be working. And what we see here is a schematic diagram of the active catalyst. I'm going to talk about that in detail. This I'm going to come back to in a moment. So we're just going to concentrate on what do we think might be happening with regards to the Fischer-Tropsch catalyst. And this is what we believe is a representation, a schematic representation of the working Fischer-Tropsch catalyst that relates to all of these wonderful products that are useful to society. We start out with a hematite structure, and the hematite structure is slowly reduced to form magnetite, and then from forming magnetite under reducing conditions, it slowly forms carbides. And one of the principal carbides is a Hag carbide, and we believe the Hag carbide to be the active phase of the catalyst. But simultaneously, it also forms a hydrocarbonaceous overlayer, which we believe affects the actual chemistry. And then in addition to that, there's a quite a large amount of amorphous carbon is built up. And so what we're seeing here, we're seeing a core, which has not been fully reduced. So this is an Fe3O4 core. Outside of that, it builds up this active phase of a carbide. And one of those principal carbides is a Hag carbide, and that's the active phase. And then over and above the active phase, there is this hydrocarbonaceous overlay. And this is what we measure in the INS. And this is actually templates this active site. It actually templates the active sites. I'll talk about that in a moment. And then beyond that, there's this amorphous carbon, which the materials have to percolate into and out of. And I've tried to describe it really within different domains. It's dependent on different hydrogen concentrations. But here, the sort of gray area is the Hag carbide. This is the active phase. And if this would dissociate CO and H2 from the syngas. And so what we do here, but the hydrocarbonaceous overload is defining access to certain sites. And so in here, we have dissociative adsorption of the hydrogen, which is then permeated through the overlayer. And here we have dissociative adsorption of the carbon monoxide, which is fed in from hydrogen. And this then goes off to form the saturated compounds. If you operate under hydrogen lean conditions, there's insufficient hydrogen supply and you just form this amorphous carbon. So between these two phases, we believe that we can understand the role of how the ion is doing all this complex chemistry. And just to reiterate, it's the hydrocarbonaceous overlayer that is defining the form of the active site, which it's essentially masking aspects of this Hag carbide. Okay, so that's our suggestion for what is happening. And what we're suggesting is that this hydrocarbonaceous overlay, which is this in purple or mauve, that's actually responsible for controlling the hydrogen supply. And what this figure is trying to suggest, that it's the control of the hydrogen supply that moderates the distribution of the products. In a rich hydrogen supply, you would see saturation of hydrocarbons. So this is forming a chain propagation reaction. So this also leads into something else that's very current. I believe in South Africa you call this chemical FTS. If you control the overlay such that you can control the extent of the hydrogen, what you can do, you can moderate the extent of the hydrogen association with at the same time with the CC propagation reactions. And rather than forming saturated hydrocarbons, you can actually form olefins. And so this is a way of being able to use the FTS method with particularly doped samples to accentuate a reaction that allows you to get very, very valuable feedstock chemicals. Okay, so this is for the future for us. We're interested in Fischer-Tropsch to olefin catalysis as well as classical Fischer-Tropsch synthesis. So I think that's okay. So I thought that would be relating to the South African colleagues. And I apologize for my lateness and tardiness, but hopefully there's some connection. You can see how the INS has provided this unique handle. And off the top of my head, there's very few methods that you could use to follow the hydrogen. There's plenty of ways to look at the carbon and the iron, but not so much the hydrogen, and the neutrons allow us to do that. So by the look at my watch, I've got 15 minutes left, and I thought I would broaden the discussion, or maybe 10 minutes left. I would like to broaden the discussion to show you another application of INS as applied to real world chemistry. And I emphasize in the investment. I understand that if you've got to access neutron sources, you've got to invest in them. And I'm going to suggest that if you do that, you can get a return back to the central economy. And I'm going to illustrate this with, this was a collaboration that was done in collaboration with Sasol Technology United Kingdom, who were operating Fischer-Tropsch processes. I'm now going to switch across to a collaboration that's very active with Johnson Matthey in the United Kingdom, and where we're looking at a certain type of zeolite catalysis using neutrons. So hopefully I have now stayed my welcome. I'm now going to talk about the methanol to hydrocarbon reaction over ZSM5. And this is actually very big in China. It's not so developed around the world. It's developed in the Middle East. And what it is, it's a route by which you can take biomass, so any form of biomass, you produce syngas, and then you use the methanol synthesis catalyst, which is a very classical process. You make methanol, and from methanol, there's this process called the methanol to hydrocarbon reaction, where you can make a whole series of basically, you can make gasoline, you can make diesel, you can make airplane fuel, and you can also make chemicals as well. So there's a process called methanol to olefins, which MTO, which is a standalone. And all of this aspect of the catalysis is done using zeolites, using solid acids. So just on the right, I previously showed you a picture of the Sasol complex in Sasselberg. This is a picture from the Middle East. It's a methanol to hydrocarbon plant owned by Exxon. And here, this is where there's an opportunity to start to make gasoline from any source of biomass. So this was a collaboration with Johnson Matthey and the Catalysis Hub in the United Kingdom, also funded by the EPSRC. And these wonderful colleagues, this is Stuart Parker, who you'll see features in all of this work, two wonderful PhD students, Professor Russell Howe from the University of Aberdeen, and Paul Collier was our contact into Johnson Matthey. Okay, Johnson Matthey, I believe, have a place in Johannesburg and in different places throughout South Africa. So methanol to hydrocarbon. It's actually quite complicated chemistry. It's been around for a while, but there's aspects of it are really not very well understood. Here it's thought to operate by a concept called the hydrocarbon pool. The hydrocarbon pool mechanism is illustrated here. It's a two-cycle mechanism. The initial cycle is alkene mechanism, and then it crosses across to an alkene mechanism, an arene mechanism. And here we're starting to see the production of short-chain alkenes. And here we're starting to see the production of methylated aromatics. And here, what I think it's here where we start, we put in methanol, we chuck out some olefins, it goes round again, and we get some complexation. And at a certain stage, it crosses over, we get a hydrogen transfer, we start to produce some alkanes, and we start to get formation of aromatic species, and then the methylation of those continues, and this is how you build up the complexity. And so what we're going to do, we're going to look at it at conditioning phase at low temperature. We're going to look at it steady state operation at 350. So that's the one we should all be interested in. But anybody that's got any interest in catalysis will know that we always have to consider a deactivation phase. And so we're going to essentially manage a deactivation phase by running the reaction at 400 degrees. Okay, so I'll just quickly go through this as just an illustration. The reaction profile, it's actually quite complicated. It needs some catalyst conditioning. This is the period, this is the initial period, this is 1 day, this is 2 days, this is 3 days on stream. This is all recorded with an inline mass spectrometer at ICIS. And here you see this rather complex catalyst conditioning period. And thereafter, we achieve steady state operation. And this is done at 350 degrees. And here we see steady state operation with effectively no deactivation. And what we see, the product distribution is exactly what we would expect. We start to see some aromatics. The aromatics is 91. That's the with regards to the tropylium ion and that. We see some dimethyl ether as an intermediate in the early stages. We start to see some short chain alkanes, and then we start to see a buildup of aromatics as a function of time. This is all in the gas phase. If we actually look at the condensate, we have a catch pot, and it's a rather poor figure, but this is a GCMS of the catch pot. And what we see toluene, we see methyl benzenes, we see trimethyl benzenes, tetramethyl benzenes, and we see a small series of substituted naphthalenes. We tend not to see anything bigger than naphthalines due to the pore size limitation of the zeolite. Okay, so this is classic MTH chemistry. This just means that our catalyst, which is a commercial catalyst, is replicating everything that's in the literature. And then really what we want to do is can we understand a little bit more about the mechanism by which this process operates. And so there's some issues that in principle in the past people have used infrared spectroscopy, and infrared spectroscopy in the early stages of reaction is okay. It tells us about the Bronsted acid sites. So these are the Bronsted, it's a solid acid catalyst. It tells us about the Bronsted acid sites that's doing the work. Here, this is the unreacted catalyst, and it's telling us that we're identifying the Bronsted acid sites here. Here we have extra framework aluminium, and here we have cyanyl groups. And it's the critical thing is really the Bronsted acid sites that's doing the chemistry. And what you can see is we increase the temperature of reaction, we start to see the quality of the infrared spectrum deteriorate. So there's a little bit of a change in the surface, but it's not so bad. But if we focus, if I just go into the that was a narrow scan. If I look at the process over an extended period of time, so this is the blank ZSM5, this is our steady state operation after 110 hours, and this is a deactivation phase. If you look at the region where we want to look at the Bronsted acidity, you can see that after a while, the catalyst is just absorbing all the infrared and we can't get any information out. And so this is a consequence of the processes of the catalysis with increasing carbon laydown. And it means that we can't, by using optical spectroscopy, we can no longer follow the process after a reasonable period of time on stream. And a common and limiting condition, and this we've classically seen with heterogeneous catalysts. So this is not unusual, but it's rather vivid in this scenario, whereas this is the species we would really like to get a handle on. And of course, on an active catalyst, we've been denied that by infrared spectroscopy. So in contrast, and this is what hopefully this you're seeing the benefit, this is the neutron spectrum. This is inelastic neutron spectra. This on the A, this is recorded on Tosca. This is recorded on the MAP spectrometer. These are two inelastic spectrometers available at the ISIS facility. And here, this is our conditioning phase, the state 1, the yellow. The red is our steady state operation, and our deactivation regime is catalyst is in blue. And so what there are, you can see this is between 0 and 2000 has been recorded on the Tosca spectrometer. The MAP spectrometer allows us to extend that range from 2000 all the way up to 4000 wave numbers. So we can cover the full vibrational spectrum. And what you can clearly see is a difference, although there's just a progressive change in the surge stretching region, we are clearly seeing difference in the chemistry of what is actually the hydrocarbon overlayer, the hydrocarbon pool that's responsible for the chemistry in this case. And what the challenge in front of us, and to cut along, this was really quite a challenge. It was very nice to be able to get this information, but this was very difficult to do. We had to discern what was the candidate molecules that were responsible for this. And in fact, the database for this type of work is very limited. And so because of that, we had to do a whole host of work where we looked at candidate molecules to build up our own database. And what this is involving simulations, but also measured spectra. And from this, we were able to, it turns out that the methyl torsion, so the methanol in this process leads to methylated aromatics. The methyl torsion is very diagnostic, it turns out, surprisingly so. And this led to a standalone publication that allowed us to build a database for methylated aromatics. And from there, could we discern which class of aromatics were responsible for our hydrocarbon pool? And this work was published in ChemCat Chem. And what it is the spectra there, the INS spectra. If I just go to the left-hand side, we've got the INS spectrum that we've seen previously. It's actually, I think it's just for the steady state spectrum. And then what we've done, we've been able to simulate and work up what the individual components of that spectrum are made up of. And it turns out to be really quite complicated. It's clearly a combination of polycyclics, of naphthalene, of ampheline. We've got specific methylated benzene rings. And what it is, at different phases of the reaction coordinate, it's forming these different types of molecule in the hydrocarbon pool, which are then controlling, allowing the catalyst to operate in which of the cycles it favors. So is it going to favor methylated aromatics? And you can actually see that the form of the hydrocarbon pool is facilitating that transfer. So this, I think, is the first time it's ever been seen. It's been postulated by NMR, but not to this degree. And this is actually came off a working catalyst. So here, what we've done in color, we've suggested what are the relevant contributors that come across. Clearly, it's a complex case, but we can actually see who are the partners and the players in this level of chemistry. So I'd just like to summarize this with the MTH chemistry. The hydrocarbon pool concept is well established, but details on the form and the molecularity of the hydrocarbon pool were previously poorly defined. An INS database of candidate hydrocarbon pool molecules were constructed, and then the INS spectra of the ZSM5 and the MTH catalysis were acquired for the three regimes: the conditioning phase, the steady state operation, and the deactivation phase. And the form of the hydrocarbon pool is now more precisely defined. We can follow it at any part of the reaction coordinate. We also have preliminary information on how these modifications to the hydrocarbon pool can influence product distribution. And what I'm saying here is that we now have a spectroscopic handle that we can actually look at sample history or sample pre-treatments, and can we modify it? Can we treat a sample such that under certain conditions, the hydrocarbon pool is so defined? And then that hydrocarbon pool will then influence selectivity. So this is an indirect route to product control. And it's clear from this process that the ZSM5 MTH chemistry is a very dynamic phenomenon. And just what we're doing now, just to show that we've built up these relationships with the company, because we've now got this new spectroscopic insight into zeolite catalysis, we're now looking at that to help develop, in partnership with Johnson Matthey, a new generation of zeolites that hopefully will exhibit minimal deactivation. And then I think this is my last slide. I thought I'd try and summarize. I know this meeting will have political connotations. I thought I would just make this comment from a sort of scientific bench, was that the investigation of heterogeneous catalysts via application of neutron scattering techniques is a growing area of scientific endeavor that is shedding new light on the functionality of a wide variety of heterogeneously catalyzed reaction systems. I truly believe that to be the case. I also noticed what's in the picture there is an award that I received last year from the STFC Science Board, and this was an impact award for economy. And I'd just like to say that this has direct links to technology and to the Chemical Manufacturing Centre, which in the United Kingdom we value very highly. And it's an investment in science, which ultimately I think is paying back to the overall economy. So with that, and I apologise for my late start, I'd like to terminate my talk. Thank you. Oh, yeah, no, that's me done. Yeah, thank you.
Thank you, Prof. Lennon, for the interesting talk. I'm going to open the floor for any questions. Are there any questions for Prof. Lennon? I know you're probably hungry, but we'll break soon. One question from me. I've noticed that the spectra looks quite noisy. The in-spectra look quite noisy. What's the reason for that? Is it just because of the amount of sample you analyzed?
Yeah, good point. Yeah, I mean, I think that I should have probably also say that neutron scattering, I don't know what, sorry, I apologize for missing Mark's talk, but neutrons, it's really quite difficult. It's not easy. And we are in an inherent, neutron scattering for us for vibrational spectroscopy is inherently weak. So if we do inelastic neutron, for diffraction, I'm sure that's not the problem. But for inelastic neutron scattering, we're in a low signal to noise regime. And those spectra are probably the best that we can get at the moment. I should say we've got one of the best facilities in the world. That they are probably state of the art that we can get at the moment. How we get around that, we use large amounts of samples. So it actually favors industrial samples because we need quite large amounts of samples. So the scattering from the last lot you've seen, I think that requires around about 12 grams of sample. And also the scan times take around about 6 hours, 6 to 8 hours. So that I concede the spectra compared to optical spectroscopy. Absolutely, it's in terms of signal to noise and sensitivity, it's a poor cousin. Yeah, fair comment.
Thank you. Any question? Yitesh? You might have to speak.
Hello, Prof. Lennon.
Yeah, hi there.
Okay, sure. Thanks, it's Chitesh Kesho here from the department. I just want to ask a question. One of your spectra, you've got 1,000, a magnification of 1,380 versus the other two spectra that were at ×2 and ×1.3. Can you maybe explain what that 1,380 represents? Is it a magnification in terms of just the vertical scale, or was it the time of acquisition?
I think that will be a frequency. I think that's a frequency, a marker of the frequency.
Okay.
We do expand some of the, really relating to the last question, that sometimes we have to expand the spectra, but we would never expand it, I think, to that extent. By the sound of that difference there, I think that I could. Do you know which figure it was, please?
It's the blue. It's the 3 hours, 440 hours and 300 hour.
Okay. If I can. Sorry, I'm not sure. This is. Let me find. Is it this guy?
No, down more.
This chap?
Down.
Oh.
No, I think you were right. Just there. Yes. Mm-hmm.
Oh, here?
Yeah. No. There, yep, on the far right.
Oh, I see. Yeah. Oh,
right.
Golly. Right. Yeah. Oh, yeah. Yes, I see what you mean. Oh, I think that's a mistake. Oh, sorry. I think that's ×13.
Okay.
Sorry, I apologize. Yeah, you're well spotted. Sorry. Yeah. So it's ×1, ×2, and ×13. There's no way it would be. Oh, is it? No, it can't. No, I can't believe it's that large. If it's possible, that means then that, oh, hang on. Oh, hang on. Maybe it is. Hang on. Okay, right. I'm not sure. I'm uncertain. You've spotted something, and what it is, that's a good, well spotted. It is possible for the deactivated state. So for the blue spectrum, that it's lost. It's in a deactivated regime. So it's starting to become carbonaceous. And if it's becoming carbonaceous, the hydrogen concentration is reduced. And so as a consequence of that, the neutron spectrum is massively reduced because it's only really seeing the hydrogen. I'm a little bit embarrassed because it looks as though one's been clipped off on the other one, but it is possible that reflects the extent of the dehydrogenation of the overlayer due to being in a deactivation regime. So it's actually a very good question. So what you're seeing is the dehydrogenation, I think. I need to check that number. I'm slightly embarrassed because I don't know what that really is. But it is possible that we are seeing the deactivation process vivid in front of us. So it's due to the loss of hydrogen in the catalyst matrix. Yeah. I can't comprehensively, I'm going to have to go back and check that. I've made a mistake in that. I'm not sure of that number. Good question.
One last question. Are there any questions from the chat box? Oh.
I have a question with regard to your model, which shows us a core of magnetite, then a Hag carbide, and then carbon. Now, you go from an oxide to a carbide.
Yeah.
Isn't there a metal somewhere?
Ah, yeah.
Iron 0.
Yeah, really good question. So actually, I think this is an amazing scenario. So what if you leave it long enough, it will go to iron zero. But if you go to iron zero, the idea is not to fully reduce the metal. So ultimately, it's working towards that. Once you get to iron zero, it starts to form almost very large. Well, it starts to form very large amounts of amorphous carbon. So in fact, what you're referring to is actually key. I'm not sure if I can switch it. Yeah. So if I can just use the mouse, what actually happens here is there's an equilibrium. There's a dynamic phase between the oxide and the carbide, and that's actually seen to be an increasing. We're looking at that at the moment. So there's a whole sequence where you start out with the hematite, you go to magnetite, you first form cementite, which is a form of carbide, and then you form the Hag carbide. And we believe it's the Hag carbide that's the active phase. And overall, if you leave it in highly reducing environment, we will get iron zero. So if we pre-treat the catalyst with hydrogen, we get iron zero. We do get metallic iron, but then we get overwhelmed with the quantities of amorphous carbon. So yeah, that's a very technical question. And how the people and they operate the plant, I think they're keeping this balance between this equilibrium between the carbides and the oxides. Yeah. Very pertinent question.
All right.
Thank you.
I think we can, is there any more question, last question? Okay. In the absence of any questions, I think we can thank both the speakers for this session.
And I must apologize for my lateness. I'm sorry. I got my clocks mixed up.
Thank you, Prof. Lennon, for your time. So I understand what will be next is to have a photo shoot. Apparently, we'll take the photo in front of this building. And I've been told that you are requested to put on your caps for the photo shoot. So those who've got their caps, I can put them on. And from there, we will go for lunch. I don't know where that is, but I'm sure the organizers will guide us where lunch is. So thank you very much for your participation. We'll see you in the next session. Thank you.
Hi there. Should I try to share the presentation? Hello. I can't hear you. Still.
Can you hear me?
Yes, now I can.
Can you hear me?
Yes. Yeah.
Are you sure you can hear me?
Yes.
All right. Okay. No, thanks.
Can you hear me?
Please show your screen. I think you are muted or you're not.
No, I'm
Please
not.
share your screen.
Let's see. Yes. Here we go. Whoopsies. Hang on.
It looks like you're no, she's not muted. She's not muted. No, she is. The screen is okay. Can you move it to that side? Let's see. Oh, okay. Or in terms of the screen, we are okay, but
Yes.
I can hardly hear you.
Oh.
I don't know whether maybe your volume is low that side or
No, it's normal.
The volume is coming okay through, yeah.
You can hear it through the remote participants can hear it.
yes.
Yeah,
yes.
we can hear you clearly.
Thanks, so we can hear you. We are ready to go. Thanks very much.
Yeah, so it's okay.
All right, enjoy lunch.
Yes, you too.
Okay, cheers.
Thanks.
Bye.
Testing.
But these people are there. So this is you.
Where
This
am I?
one.
It's usually for. Now get also to do that for the slide.
But the slide should be shown by. Shouldn't it be shown by the example?
Not gonna hold in slide.
Oh, that one.
Hi Zoe, are you there?
Yes, I am here. Do you need me to share my screen?
You, oh, thank you. We are back.
Can you hear me?
Can you speak?
Yes, I am speaking. Can you hear me?
Yes. Thank you very much. Yeah.
Okay.
We just waiting for the delegates to assemble.
Okay.
And yeah, we can see your screen as well.
Super.
Yeah.
Maybe let me stay. Yeah, I just show you mutate. Yeah, I'm looking at the participants. My girl, only was yeah. Those ya kila microphone muted.
Good afternoon, everyone. We are a little bit late, so I think we should start now. My name is Robert Chimirimana. I'm an instrument scientist from the imaging group at Nexa. I'd like to welcome those just joined us, especially those watching us from Zoom and YouTube. They're following us virtually. We had 20 delegates who were following us virtually in the morning. Hopefully they join us again. So this first afternoon sessions, we start with 2 presentations following the imaging application that we had this morning. So the first application will be in life science. The second one will be energy storage. The first presenter is Dr. Joey Fisher. Fisher completed her undergraduate studies in the University of Stellenbosch in 2000 before moving to USA. After completing her studies, she went to University of Florida in 2006. Later, she joined the Los Alamos National Laboratory in New Mexico. Now she's involved in deuteration and macromolecules characterization teams at ESS. Up to you, Dr. Joey.
Thank you very much. And thanks to Andrew for the invitation. And I wish I was there in person. It would be nice to see some of you again. So yes, today I will talk about life sciences and neutron scattering, some of the possibilities, but also some of the considerations that are maybe a little bit special to biology. So first we'll chat a bit about neutrons as a tool in life science research, then the value of isotope labeling and why it is very important for biological materials. I'll briefly mention the different techniques that are most used in life science research with neutrons. And then I will go to 2 very specific applied scientific examples. One where neutron protein crystallography was used, and the other small angle neutron scattering. And then finally, I just have a couple of slides on ESS support for samples and labs. And then hopefully we have a few minutes for Q&A. So what you also heard this morning from Andrew is that neutrons are today, but also historically, underrepresented as a research tool in the life sciences. But I would say, from my perspective, the last 5 to 10 years, this is changing. And I feel the field is really growing in the last years. There are always the same issues that are held up as the reason for this kind of bottleneck for not having so much expansion into the life sciences. And the biggest thing that doesn't just affect us, but it does affect us, is that the neutron flux is inherently low, and we really suffer from bad signal to noise with biological samples. They're complex, they have lots and lots of atoms, and yes, they are hydrogen rich. So signal to noise coupled with low flux can be a real complication to getting good quality data. And all of this, of course, has a knock-on effect on sample requirements. And not only do we need to isotopically label the materials, which requires some technical skill and also quite some cost, but there's also, for some kinds of interesting materials, they are just naturally low abundance in nature. So to get enough, let's say, of a membrane protein, you may only be able to get micrograms out of a single preparation, and you need gram amounts for neutron measurements. There can be inherent bottlenecks that are sometimes you're not able to overcome. Then there are a few dedicated instruments that are really focused and specialized for life science research. And of course, typically science is coming from biology, medical doctors, microbiologists. They are absolutely not experts in neutron scattering methods, and they are not expert users of large scale facilities. So to get more of these people in, we really need facility staff to support the entire pipeline, from sample, understanding what sample you need, to preparing the sample, and then really helping with data analysis as well as interpretation. However, all these issues, neutrons are uniquely well suited to the study of biological materials because we can have them on time and length scales that perfectly match the size of the kinds of things we want to study, whether it is crystal structures on the atomic level detail, to micellar aggregates, to viruses or bacteria, and we can actually use neutrons all the way to intact living things, such as this plant shown here. So while neutrons have properties that make them very well suited to study all kinds of materials, biologists really benefit for the following reasons. One, we already heard that neutrons are very sensitive to hydrogen atoms, and we'll talk more about that soon. The methods are highly complementary to the other things we already tend to use in the lab. You can, of course, X-ray scattering techniques are common, but they can be complementary to microscopy, fluorescence measurements, UV vis, and so on. So there is a neutron flavor in terms of energy that can match what you are already doing. Generally, in the energy range we use for biology, they are non-destructive, which is an enormous benefit. And it means that we can study biological materials closer to physiological temperature, for example, at room temperature. We don't have to freeze everything. We can look at things more in their native state. And there is no ionizing radiation. So in the field of metalloproteins, for example, where you really suffer from oxidative damage to the metal centers, you can completely avoid this artifact and complication by using neutrons, for example. And like I said, the time and length scales are very well matched to biomolecules. However, it is maybe good and bad that hydrogen is abundant in biological materials. They make up something like 50% by atom count of the atoms that compose proteins, DNA, carbohydrates, and so on. But they are abundant, perhaps because they are so important, and they are involved in every physiological process that sustains life, essentially. Whether it is hydrogen bonds, protein folding and function and structure, to enzyme activity in terms of catalysis, and of course, hydrogens are very much involved with ligand or drug binding to medically relevant targets. So the fact that they are everywhere is a complication, but it's also an opportunity, let's say, to study what they are doing. So I'm just showing this very biologist reductionist view of the periodic table, where these are the atom types we typically deal with in the materials we study. And I won't spend too much time except to highlight again that it is the ability of neutrons to distinguish between isotopes of the same element. And the fact that this is especially true for hydrogen makes it such a good probe for biological materials. And you know that the scattering properties of the isotope deuterium are very much superior to those of the more abundant protium isotope of hydrogen. And for this very reason, circled in red, it is very beneficial to deuterate the samples. And deuteration, we mean replacing the naturally abundant hydrogen with as many deuteriums as we can. And so you can partially deuterate your samples, or you can fully deuterate them. Then we tend to talk about per-deuteration, or you can also do HD exchange, where you only are able to get rid of the labile or accessible hydrogens through a simple exchange process. And this is something we have to do to biological samples. And this is something my group at ESS does. And so there are different kinds of deuteration. And even though both involve deuterium, they are very, very different. It requires different kinds of labs, different equipment, and also different training and expertise. For chemical deuteration, we do organic synthesis of small molecules, where you either purchase deuterated precursors and solvents, or you can actually make some of these molecules yourself by using a PAR reactor. And here we do catalyzed HD exchange under elevated pressure and temperature. And in our group, for example, we can make a number of what myself as a biologist would call small molecules, whether it is detergents like DDM, phospholipids like POPC, or truly small molecules like pyruvate or lactic acid. Biodeuteration is a completely different beast. Here, instead, we rely on living cells to produce for us the deuterated materials. And the trick here is to adapt and adjust the cells to growing in heavy water. And in fact, many species are able to do so reasonably happy. So yeast we can culture under fully deuterated conditions, Then we can process these deuterated cells and extract valuable products such as phospholipids. We can do the same with E. coli, which is bacteria, where we can grow it under deuterated conditions and then extract proteins or DNA that are labeled. And finally, my group, we also grow algae in heavy water. And these we essentially cook or boil to make a rich cell extract that we can then feed back to E. coli as a source of deuterated nutrients. So here, really, you're just preparing a very simple cell media in heavy water, supplemented with a deuterated carbon source. Then the cells do essentially all the work for you, and the skill comes into extracting and separating the molecules of interest. The purpose of deuteration very much depends on the technique you want to use. So on the left-hand side here are shown examples from protein crystallography using neutrons, where we have to deuterate to really reduce the background. How do you say, the background that we contribute to the presence of hydrogen. But we are also interested in the actual atomic position of the hydrogen, because we would like to see how water molecules, for example, form networks. We would like to see hydrogen bond donor acceptor groups. And the huge power of neutrons here, coupled with deuteration, is that we can distinguish between neutral and charged amino acid residues. And in this case, the lysine is in fact catalytically important for an enzyme. And us knowing whether it's neutral or charged can have a big effect on our understanding of the basic mechanism of an enzyme, for example. So here we try to boost the signal to noise, get rid of the background, but also actually observe the hydrogen itself. With other scattering techniques, for these kind of materials, we use a concept called contrast variation. So here you are selectively deuterating components in a complex. Then you take your neutron measurements in mixtures of ordinary water and D2O. And at some point, you can actually get what is called contrast match out. And this is a point in the mixture, which is a combination of the H2O D2O mixture and the level of deuteration in your molecule that makes the molecule effectively invisible. It doesn't contribute at all to your neutron scattering signal. So a common example that is used to illustrate this concept of contrast variation is shown on the bottom here, where you are measuring, say, a complex of protein RNA in water, and then as you increase the D2O, the deuterated protein part becomes invisible, and you can only see one of the components in this complex. And this is a very important concept that is relevant for reflectometry and SENS specifically. So the most used neutron techniques for life science, in no particular order, is typically small angle neutron scattering, reflectometry. We do single crystal diffraction for neutron protein crystallography, and imaging. Imaging is probably the least developed, but it is quite used for biological samples that relate to intact living plants. And it really is a shame because neutrons have such high penetrating power, and they don't immediately kill the plants due to radiation damage, which means you can actually see into pots and soil due to this penetrating power of neutrons, and actually observe what the plant roots in this case are doing. In this study, they actually watered the plants with D2O, and then they were able to follow the uptake into the root system, but also how the water moves through the soil. And this has, of course, implications for water use, how to be more efficient with agriculture and crop production and so on. So a lot of the imaging examples that look at biologically relevant systems is, in fact, on plants. There are a few studies that I'm aware of that are looking at bone implants, where, again, you can image better the biological bone material than the implant material that is typically made of metal and quite transparent to neutrons, depending on the material, of course. Reflectometry is a slightly higher resolution technique, but here we are looking on thin films and interfaces in this nanometer range resolution. And this technique can give us information about the internal organization of membranes. And shown here, for example, is a biological cell membrane, a double bilayer. And we can learn things about the thickness, how even it is, and even the composition. And since these are wet systems, you can also actually easily change the conditions around these materials that mimic where they are located, let's say in the cell or the body. Contrast matching is also heavily used here, and deuteration is very important to be able to get meaningful neutron reflectivity curves out of biosamples. And here you can imagine that there are actually a lot of like antifungal peptide drugs that are known to be membrane disruptors. And there you can study the disruption and changes in the membrane upon addition of a therapeutic agent. Crystallography works the same way as X-ray crystallography, just with neutrons. It's single crystal Bragg diffraction, and the goal is here to determine the atomic crystal structure of protein molecules. The resolution we aim for is usually 2.5 angstrom or better, and like I explained before, the deuteration is really to reduce the background boost signal to noise, but actually also to determine the location of H and D atoms in the structure. If we are able to do this, we can actually see many features in protein crystal structures that tell us about how they work, and also maybe how substrates or ligands can bind to them. Small angle neutron scattering is perhaps a bit lower resolution than crystallography. Here again, we're looking in the 10 to hundreds of nanometer range typically. And for biosystems, this is a very powerful technique. It is very well suited to looking complicated, flexible dynamic systems in solution, which perfectly describes many large protein-protein complexes, for example. Typically also things that are not well behaved enough to crystallize, you can say, or are perhaps even too big. And again, here, like reflectometry, the concept of contrast matching and deuteration, strategic deuteration is heavily used to be able to make sense of SANS curves. And we can get information about the size, the structure, and the behavior of molecules in solution. Now to the two examples that I think are very near and dear to every human on the planet. I will tell you about coronavirus studies using neutron protein crystallography of the main protease. That was done by a group of scientists at Oak Ridge National Lab. And the second example relates to small angle neutron scattering, looking at lipid nanoparticles encasing mRNA for therapeutics. And here, of course, you can immediately think of the Pfizer or Moderna mRNA-based vaccine against coronavirus. So the coronavirus genomes are translated into these very large, complicated polyproteins that are not functional when they are made. These need to be processed into functional protein units, and the virus does that by using one of its own proteases called the chymotrypsin-like protease, or the main protease, MPro. And here is a ribbon diagram shown of this viral enzyme. It is active as a 34 kilodalton homodimer, and it was very early on recognized that the activity of this particular viral enzyme is essential and required for viral replication, which of course, unsurprisingly, makes it a very, very good drug target. So since the beginning of this pandemic, there have been a lot of efforts to look at repurposing existing approved drugs that are used in the clinic. And some of these studies focused on looking at HIV and hepatitis protease inhibitors with quite some success, actually. It turns out that these protease enzymes in viruses have very similar activities in active sites, and there's actually a lot of cross-binding of these inhibitors also to the coronavirus protease. So yes, a lot of screening efforts revealed that some of these are actually decent inhibitors also for coronavirus. And the main efforts have been focused on finding either reversible or irreversible covalent inhibitors that can bind and ultimately jam up the active site of this enzyme, thus being able to stop viral replication, of course. On the right-hand side is a very nice structure of the main protease active site with some of the important amino acid residues labeled. I'll just point out cysteine 145 is the residue that is covalently modified when inhibitor is bound. And the protonation state of the histidine residue surrounding the cysteine, as well as the electrostatic state of cysteine itself, can tell us a lot about not only how these enzymes work, but also how we can, exploit this information to better inhibit them. And to be able to do these studies, we really need, beyond just what X-ray crystal structures can give us, we need atomic level information on the electrostatic state or charge distribution in the active site, and also information on the hydrogen bonds and the solvent, the water molecules, and how they mediate or are involved with ligand binding. So that's exactly what this group at Oak Ridge did. They prepared crystals, protein crystals of this main protease by itself, that is shown in pink here, the APO enzyme, and in complex with a hepatitis C inhibitor called teleprevir. And then they did very, very careful neutron structure determination of both, and were able to do a side-by-side comparison and analysis. And this involved careful mapping of the hydrogen atoms in the active site with and without this inhibitor bound, and then be able to infer information on how the charges change and shift in the active site to accommodate drug binding, or how that happens because of drug binding. It's the chicken and egg thing. It's maybe not easy to know which affects which. So here's a nice summary, table 1 from their paper, that shows that they were able to do this comparison, the ligand-free versus teleprevir complex, and they were able to observe that the net charge of +1 is actually conserved between these two forms of the main protease, but that there are a series of electrostatic changes happening in the histidine residues that surround this catalytic cysteine residue. And this was a very interesting observation, and shows kind of how flexible enzymes can be and how much they are able to do to accommodate an inhibitor binding. So in this case, neutron protein crystallography really gave unique information, the kind of information on drug binding you cannot get with any other method. Enzymes like the main protease rely on the transfer and movement of hydrogen atoms and water molecules, and careful investigation of these events actually allows us to see where they are and then help us understand what they are doing. And ultimately, in this case, the goal is, of course, to see maybe why existing drugs work, but also to inform medicinal chemists on how they can improve these inhibitors, or maybe make them more specific to this kind of protease. The second example is very much focused on small angle neutron scattering, and it was instrumental in understanding lipid nanoparticles that are used to deliver mRNA in certain drugs or therapies. The development of RNA-based therapies has been around for quite some time, and these tend to use LNPs, or lipid nanoparticles, and there has been a lot of progress in this field in the last years, not least of all due to pressure to develop vaccines in a very rapid way in response to the coronavirus pandemic. Today, there are several approved therapies in use, some like the mRNA-based vaccines against coronavirus, but there are also therapies being used employing small interfering RNAs. They're smaller than mRNAs, and these target transthyretin, which is a protein suspected to be involved with Alzheimer's, but also cardiac amyloidosis. And a critical component here is the lipid nanoparticle, of course. And a lot of work is going into understanding the components and how these ultimately affect the size and structural features of these LNPs, because these, of course, have some effect, how they are distributed in the body and how they are taken up as well. It has been known for some time that this apolipoprotein E interacts with these LNPs. Ordinarily, ApoE is found in our serum and is responsible for lipid trafficking in the body under normal physiological conditions. But it has been shown that these love to bind to these LNPs when they are injected or introduced into the body. And when ApoE absorbs or coats these particles, they tend to be shunted and preferentially directed to the liver. So a lot of these mRNAs or siRNAs that are delivered in LNPs, when ApoE binds, they get directed to the liver, which for some things may be preferable, but for other applications, maybe that is not the primary site where you would like your mRNAs delivered in a therapeutic setting. In addition, LNPs can also enter cells via other methods like endocytosis that also are influenced by the composition of the lipids that make up these LNPs. And in the case of the mRNA vaccines, for example, it wasn't known whether ApoE is required for proper trafficking and delivery of the mRNA, or if it may be interfering and making the process less efficient. So this group that are affiliated with AstraZeneca, they did really beautiful SANS studies to study really the details of the composition where all the components are in the LMP when they are assembled, and how ApoE exposure changes how these LMPs actually look. So they designed a series of really nice SANS experiments where they prepared LMPs with RNA while they were varying the amount of deuterium incorporation into the different components that make up these LMPs. And they did this so that they can do this whole concept of contrast variation to see where certain components are in the molecule, in the final product, let's say. So that's shown in the gray and black cartoon here on the bottom right of the slide. So under some solvent conditions, you can maybe see that some of these molecules are preferentially partitioned more to the shell of the LMP, whether others tend to crowd around the mRNA in the center or the core of these LMPs. And then they take these molecules and they repeat these measurements, but after they've incubated with ApoE, and then they can, of course, measure the difference or the change. And there were quite some changes as a result of being exposed to ApoE. And these bar graphs shown here are just a summary of lots and lots and lots of SANS measurements. And they were able to do volume fraction calculations, which gives quantitative information on the changes. And what they were able to see is that exposure to ApoE causes a compaction of the LMPs in addition to a redistribution of the components. So when ApoE is bound, you can see that cholesterol tends to move more towards the surface, whereas other lipids tend to go to the core. And the overall size of the molecules tend to shrink. And this, of course, has strong effects how LMPs will be rooted and absorbed in the body, and may ultimately actually degrade the efficacy of the mRNA that is being delivered. Whoops. Oh, good. So in this kind of study, SANS really is a crucial tool for drug formulation. The contrast and level of detail you can see here, of course, is not achievable with other solution scattering methods. And I think there is a lot of work to be done here, as there are essentially an infinite amount of variables when designing and composing LMPs. There is a company that is making these for drug delivery studies that have made over 500 flavors of LMPs that can be used to package different things. So yeah, there's probably a lot more work to be done to understand the composition and size and how that is a determining factor for efficacy when introduced into the body. So finally, just a few words about the ESS. So ESS is, of course, Europe's next generation spallation neutron source. And we are building 15 instruments in the first go around. And we will indeed have at least one instrument of the type I mentioned before. At the top here, you can see Odin will have an imaging instrument that will be able to look at life science or biological materials. We are also building Loki and Freya, SANS and reflectometry, and my favorite, which is the NMX beamline, which is aimed at neutron protein crystallography. However, instruments are fantastic and of course really important for being able to use neutron facilities. However, we should also not forget that users need sample and laboratory support. And the ESS is indeed investing quite some manpower and infrastructure into building very, very good labs attached to the experimental halls, and also groups like mine that are purely aimed at supporting users in chemistry, soft matter, and life science to be able to get deuterated materials. So we are already, even though we don't have neutrons and instruments in operation yet, we do since 2019 actually offer deuteration and sample prep support for neutron users that are all over the world. Of course, you don't or can't be an ESS user today, but we do make materials and offer support to the broader user community wherever you may get your neutrons. If you are curious, you can check out our user office, our new portal that we use for proposal submission and review. Access like beam time is granted based on having an accepted peer-reviewed proposal. And we just now today opened rolling access where we will offer support for biological deuteration and protein crystallization support for a limited number of proposals. And you're welcome to reach out to me or to the team if you would like to know more. So with that, I'm finished, and thank you for the invitation and thank you for the attention. And I think we have some minutes left for questions. not many, just a few.
Thank you, Dr. Zoe, for excellent presentation. Any question? Do you have any questions for Dr. Zoe? And those following us online, they can put the question in the chat Zoom. Okay, if there's no question, let's thank again Dr. Zoe.
Thank you so much.
The second presentation for this session is by Dr. Nelima Paul. She is a group leader in data evaluation group at MSZ and Technical University of Munich. Her focus is on Lion research batteries to make safer, efficient, and to prolong the lifetime of the batteries, which is very crucial for the industrial need nowadays. So Dr. Nelima Paul, up to you now.
So thank you very much for the kind introduction and the invitation to present some of my results here. I'm looking forward to it. And now I try to share my screen. Can you see my screen? Are you able to see my screen?
Yes, we can.
Okay, so, and I will now change to the pointer mode. Yeah. So my talk is, in my talk, I will try to show you how we use neutrons to answer key questions in aging, related to aging in batteries. And I will try to show you how it is possible to improve the battery performance and lifetime, and by modifying certain components within the batteries, and how neutrons and in some cases also X-rays are useful in answering key questions. So all these experiments are done at the FRM2 research reactor in Germany. So it's called FRM2 because we had the first research reactor called FRM, which was the first German neutron research reactor, which was commissioned in 1957 and stopped in 2000. And since 2004, we have the FRM2 research reactor. And this reactor is located here near Munich in the south of Germany. And our reactor is based on a single fuel element, which is very compact and highly dense. And with only a moderate thermal power of 20 megawatts, we are able to deliver a very high flux, 8 to 10 to the power 14 neutrons per centimeter square per second. We have about 30 instruments in operation. So most of some of them which use thermal neutrons are around here, located around the new reactor core. And the experiments which use cold neutrons are farther away in our guide hall. And we are also opening some, so starting up with some new instruments in future. So why neutrons are useful for energy research? So you have heard about some very nice properties about neutrons, but they are also useful for batteries and energy research, because as you can know, we can perform, you can have batteries enclosed in cryostats and in furnaces, and you can operate the batteries and come to know what is really happening within the battery, because the neutrons will really not have a strong interaction and will not harm the battery in any way. And normally in batteries, we use lithium-ion batteries, and then for lithium, which is a very, which has a very low atomic number, this can be seen very nicely with neutrons. And that is why neutron research is really important for this field. So the typical way to apply for beam time at our research reactor is you go to our website, and there are calls certain times a year. So you write a 2-page proposal describing what you want to do, what you have done in the past, and why you think neutrons are useful. So a 2-page proposal. And then if it is accepted after a review with the international committee, you are invited to perform the experiments, which take place for 2 days or 5 days, depending on which instrument you choose and how your proposal is. And after that, we have recently we have opened a new data evaluation group, so we can help you with the evaluation of your obtained data at selected instruments. This is a new facility which is open for new users or inexperienced users. So the outline of my talk would be follows. I will show you why lithium-based batteries are important, why it is important to study them or do research on them. What are the main aging mechanisms? So aging means degradation. So what are the main degradation mechanisms which take place in these batteries? And then I will show you, using selected examples and using various types of anodes, how we have improved the performance of the battery and how we have answered key questions using neutrons. So lithium batteries, as you all know, are present everywhere. So they are not only in your consumer electronics like laptops, telephones, cameras, but they are also being used in stationary energy storage units. They are also being used in all sorts of electric vehicles. However, electric vehicles are not new. They were already used 1 century ago, as you can see from the slide. But the main difference, the reason why the battery-operated cars did not progress so much was because at that time they used lead-acid type of batteries. So if you look at this graph here, it will show you how much energy can be stored per unit weight on the x-axis and per unit volume on the y-axis. So the batteries of the older generation, they used lead-based batteries, which could store only a small amount of energy. And now we use lithium-ion batteries, which can store a much higher amount of energies per unit weight and per unit volume. And this is why lithium-ion battery-operated vehicles are now more and more on the streets. In fact, this graph is quite old. We are much higher on the top right-hand side now. So to those who are not so familiar with lithium-ion batteries, maybe a small introduction. So in battery, you have typically an anode here and a cathode, and these are separated by electrolyte, which is electronically insulating and ionically conducting. Therefore, when you charge or discharge a battery, the lithium ions flow from one electrode to the other through the electrolyte, and the electrons, they flow in the outer circuit. So basically, it's a shuttling mechanism where lithium ions are exchanged. Now, because of the limited electrochemical stability window of the electrolyte, what happens is that typically solid electrolyte interface layer is formed at the electrolyte anode interface, shown here. So this passivation layer, called SEI layer, is actually useful because it stops further reduction of the electrolyte. But this SEI layer has to be really stable for a good operation of a battery. At the same time, you should have not too much of an SEI layer, because this SEI layer absorbs the electrolyte and it also absorbs lithium ions. So one of the aging mechanisms is loss of the cyclable lithium and electrolyte into the SEI layer formation and growth. The other type of aging mechanisms are structural degradation of the electrode materials, the anode or the cathode, and another degradation mechanism which causes short circuits and fires is lithium plating. This leads to formation of dendrites, and these dendrites go from one electrode to the other and can cause short circuits. So current research is focusing on various topics. For example, they are trying to replace this liquid electrolyte, which is flammable, by polymer or ceramic electrolytes. They're also trying to coat the anode materials with some artificial coatings to increase the safety. And they're also trying to optimize the electrodes. So for example, people are trying to replace the graphite electrode, which is conventionally used, by silicon or lithium metal entirely, or they are trying to put some silicon and prepare a silicon graphite composite alloy as an anode to increase the energy density. So in my first example, I will show you how we were able to increase the lifetime of a battery by changing the morphology of the graphite anode. And we found out what are the major aging mechanisms using the technique of neutron diffraction. And we used a full cylindrical battery, which looks like this. So before we proceed further, I'd like you to know that when we have pure graphite, this is how it looks like. And you have one peak which corresponds to graphite in your neutron diffraction data. But if graphite is half filled with lithium ions, then this graphite peak disappears and you have this LIC12 diffraction peak in your neutron diffraction data. And if your graphite is completely filled with lithium, then you have a peak of LIC6 in your diffraction data. So this is important to know for the rest of the talk. So what we did was we prepared, so this work was done in collaboration with battery company Varta. And what they did was they prepared for us two identical cells, which were identical in every way. So they had the same electrolyte, same cathode, same type of anode, except that in one anode they used graphite, which had flaky shaped graphite particles. It was called needle coke. And in the other case, they prepared graphite anode, which contained spherical shaped graphite particles. And as you can see here from the graph of capacity versus cycle number or time, the cell which contained this spherical type of particle, it decayed much slowly in capacity, so it aged very slowly, whereas the cell which contained the flaky type of graphite, it aged very fast. So they were not able to find out why this occurred, although the lifetime is improved, but the reason for the improvement of lifetime was not clear. So they came to us to answer the question. So we did neutron diffraction experiments and tried to answer these questions. So we measured neutron diffraction of these cells in the extreme state of health and extreme state of charge. We got diffraction pattern which looks like this, and we fitted these diffraction patterns using reticle refinement, and we found out a lot about the structure. So what we found out that here there was no material degradation taking place. The only aging mechanism which was taking place here for these cells was the loss of lithium into the SCI layer formation and growth, and there was no other loss mechanism. And the reason why you had the better performance in the spherical cell was the tortuosity. So when the cell was charged fast, it was easier for the lithium to go from one part of the anode to the other when the morphology was spherical, rather than when the morphology was flaky. Then the lithium ions had to travel a much larger path, and thus you saw a effectively larger capacity loss for the flaky type of cells rather than for the spherical type of electrode cells. So this was an experiment where we were able to answer a key question on the loss and how to improve the battery further for fast charging. In another example, so this was now done at a different neutron diffraction instrument in FRM2. We were now trying to see what is actually taking place in the graphite at low temperatures. So you know when you charge cells at low temperature at very fast rates, there is some lithium plating which takes place. And when lithium plating takes place, you lose some lithium in form of metallic lithium formation on top of the anode. And this is a real data which you get from our instruments. You can see how the peaks change. So there are peaks here which correspond to the carbon, graphite, and then as the cell is charged, you see this LiC12 peak forming, and then you see the LiC6 peak forming. So this is what we see. And now we try to find what is the intensity change taking place after we have charged the cell very fast, and then note how the lithium content is in the graphite after during relaxation. So we use for this a cryostat, and again we use this type of a cell. So we have in one case charged the cell very fast, and then we observe it during relaxation, and we observe this increase in the LiC6 reflection during rest, and then a decrease in this reflection after more time during rest. Whereas when we charge a cell very slowly and then observe the cell during rest, there is no change taking place in the LiC6 reflection. So what is important here to notice that when you are putting the cell at rest, you are not subjecting it to any current. So you should not see any diffusion of lithium, but we do see a change in the lithium content in the graphite. And this is because when we have charged the cell very fast, we have caused some lithium plating on the surface. And during rest, this lithium from the lithium plating is now diffusing into the graphite. And you see here, it is not just one step of relaxation, but rather two steps of relaxation. This is because this cell contained some silicon as well in the anode along with the graphite. And in the first part, the lithium from the lithium plated layer was going into the graphite. And in the second step, it was going from the graphite to the silicon. So we can not only detect lithium plating indirectly with neutron diffraction, we can also see whether it's a one-step relaxation or a two-step relaxation. And in the next, so I heard in the previous talks that there was much curiosity. So how many methods can you use for energy research? So at FRN2, we use a lot of methods, but I will only show you some methods. So for example, now this is method number 2 and method number 3, which we use for battery research. So small angle neutron scattering you're already familiar with because of talks of the previous speakers. But neutron depth profiling is a relatively new method for battery research. And here what happens is that neutron is absorbed by a lithium-6 isotope, which then gives out these charged particles of helium and triton in opposite directions of well-defined energy. And we can detect these charged particles, and we can find out from where the lithium was, from which position these charged particles originated. So we find out where the lithium was, and from the number of charged particles, we can find out how many lithium particles there were in that position. So we can have a lithium density profile as a function of depth. So lithium is just one example. You can also do depth profiles of boron, for example, with this method. For this, for energy research, lithium is more important, and I will show you this example. So the first question in this case, we wanted to know, so we know already that if you put silicon in a graphite, you can increase the capacity of the battery. But how much silicon to put? That was the question. So we prepared silicon graphite composites with different amounts of silicon, increasing from in this plot from left to right. And then we looked at the electrochemical capacity curves, and we saw that when we had a large amount of silicon, we had a large amount of capacity, whereas when we had no silicon, we had less capacity. So we succeeded in increasing the capacity by putting more silicon. But we also wanted to know what really happens. Is the addition of silicon leading to some problems? And this is why we wanted to do neutron depth profiling. And we wanted to find out what is the lithium depth profile within the anode when silicon is added to the graphite. So as you remember, I had said we also have some SEI layer formation. And SEI layer formation is not so nice if it consumes too much lithium. And we wanted to see with this example that was the addition of silicon causing an increase in the SEI amount. So this is now the data from our neutron depth profiling experiment, where we plot the lithium content as a function of depth. So depth means the electrode depth. So in this case, we looked at the electrode. So we prepared silicon graphite electrodes with different silicon contents, and we discharged them totally. And we looked at the lithium profile. And in this case, the lithium content is only from the immobile lithium in the SEI, because it is only from the discharge state of the electrode. So there is no active lithium there. And we observed that when you have more silicon in the graphite, you have a higher amount of lithium captured in your SEI layer, which is now immobilized and not available for further battery operation. But when you had no silicon, you have a less amount of lithium, which is absorbed by your SEI. So even though addition of SEI leads to an increase in your capacity, it also sort of leads to a higher immobilization of lithium in your SEI layer. The result too was we looked at the lithium content as a function of depth when we changed the depth of discharge. So this we wanted to do in order to find out whether the silicon graphite composite was completely well utilized with lithium. And this is also what we saw, that indeed the distribution of lithium was completely homogeneous throughout the entire silicon graphite composite. And there was no transport limiting phenomena taking place when you have more silicon. So this was about neutron depth profiling. And now we also looked at these silicon graphite composites using SENS. And here the purpose was to try and understand the silicon nanoparticle and its morphology. So we had silicon nanoparticles mixed in graphite to form a silicon graphite composite. Now, one disadvantage of silicon is that when a silicon particle is pristine, so uncycled, it is very small and compact. But after cycling, it becomes big, so it expands like this, and it contains pores. So this is because during lithium incorporation and lithium leaving the silicon, it causes a large amount of expansion and contraction of the silicon. So with techniques like TEM or SEM, you are able to see that these silicon particles has these pores and it has expanded, but you cannot know if this is true for the complete electrode, one thing, and it is also not sure if these pores are empty or they are filled with SEI products. And this question, to answer this question, we did deuteration experiments. So in one case, we cycled the electrode in a typical hydrogen-based electrolyte. And in the other case, we prepared deuterated electrolyte so that we have a contrast. So this is because in the first case, in the hydrogen-based EC electrolyte, the contrast was similar to that of the silicon matrix, whereas in the deuterated base, it was different than the matrix. So in this way, we cycled the cell in a deuterated electrolyte, protonated electrolyte, and then we extracted the electrode in the fully discharged state. And then we did SANS on these samples. And we found out that these pores are completely filled with SEI products. And we also found out the mean pore size was about 10 nanometers. So this was not possible by any other method to find out the pore filling and also the pore size for a large electrode volume. So then we now come to another example. So in this case, we had looked at titanium oxide anodes, and we used the technique called grazing incidence small angle neutron scattering. So what we did was we had these titanium oxide nanotubes, which are capable of incorporating lithium and lithium removal. But if we have now silicon coated on these titanium oxide nanotubes, we can have more lithium storage. And this you can see from this curve here. So when we had just titanium oxide nanotubes, we had a capacity of about less than 200 milliamp hours per gram. But when we added the silicon, we had about 360 milliamp hours per gram. So it was possible for us to increase the capacity with silicon, but it was not enough. And this was because we were not coating the silicon completely along the entire length of the nanotube, but only from the top, only at the top. And this is something which we found when we did grazing incident small angle neutron scattering with a depth resolved mode by using neutrons with different wavelengths. So the neutrons which had lower wavelength were able to penetrate deep into the electrode and look at the region here, and the neutrons which had lower wavelength were only so lower wavelength, they were able to penetrate deep into your nanotube, and with a higher wavelength, they were only able to penetrate the surface. So we found out from these side peaks, which are indicative of the distance of these nanotube pores, we found out that the silicon coating is only there on the surface and not on the entire tube, which is why the capacity is not as much as it should have been. And when our colleagues did some side view small scanning electron microscope, they found out indeed the coating was only in the top and not in the entire. So this is also some relevance of your so using different wavelengths and probing the morphology as a function of depth. And of course, you can also find out how much porosity had changed and how much the radius had reduced due to the incorporation of now the silicon onto your titanium oxide nanotube. So in the last example, I will show you how it was possible for us to increase the lithium metal anode safety and efficiency by applying coatings to the lithium metal anodes. And for this, we did not use neutrons, but we used X-rays. So lithium metal is very promising future candidate for an anode material because it can store a large amount of lithium because it's just lithium. And so you have an unlimited supply of lithium. But the problem with lithium is that it is very unstable. Therefore, as soon as you bring it into a contact with an electrolyte, you form a natural SEI layer, which is very brittle and very unstable. And it gives rise to some hot spots through which these dendrites go. And these dendrites are not good because they lead to short circuits in batteries. So if you want to use a lithium metal anode, you have to have an artificial SEI, an artificial covering. And this artificial covering layer should have some properties. So for example, it should have a good mechanical stability. So if it is very stable, this coating on your lithium metal, it will sort of, because of its mechanical stability, strength, it will sort of suppress these dendrites. And or mechanical stability is also if it is conformal. So if your polymer coating is conformal, it will adjust to the shape of your lithium deposits. So, and the other property of your artificial layer is that it should have a spatially homogeneous distribution of lithium. It should allow for spatially homogeneous distribution of lithium. And the third is that this polymer layer should have a good chemical passivation so that you avoid any sort of parasitic reactions between the electrolyte and your lithium metal. And so these are the requirements or key requirements when you use an artificial coating layer for lithium. But one question is unanswered or was unanswered, like regarding, and that was regarding the mechanical stability. Should we really have a strong coating layer, or is it better if we have a viscoelastic or soft layer? So is mechanical strength the important factor? So this question we tried to answer in our experiments. So we prepared polymer coating layers, which had the PFPE backbone. So it's called perfluoropolyether. And we used different type of I units and M units. So the I units have weak hydrogen bonds and M units have strong hydrogen bonds. So if you prepare a series of polymers having different ratios of M units and I units, you can prepare polymers which have a similar chemistry but a different mechanical strength. So for example, the polymer containing no M units, which is M0, and only I units, I1, so M0, I1, this has the lowest mechanical strength. And the polymer having only M units and no I units, which is M1, I0, this has the highest mechanical strength or should have the highest mechanical strength. So we prepared polymers in these ratios. And first of all, we wanted to confirm indeed, is the mechanical strength as it should be? Is the trend following what it is? So we performed some frequency dependent oscillatory shear measurements on all these polymers. And we found indeed that the polymer which had a lowest amount of M units had a low modulus, whereas the polymer which had a high amount of M units had a higher modulus. And moreover, if we compared the storage modulus and the loss modulus, we found that the most mechanically strong polymer also was solid-like through the entire frequency regime, whereas the polymer which had no M units was mechanically weak and also liquid-like during the entire frequency regime. So the mechanical strength was confirmed by these measurements. And then we looked at these polymers using small angle X-ray scattering. And we wanted to find out, first of all, what is the length of these I units, because you see they were of in nanometer units and it's difficult to measure it with other techniques. So this is why this was useful. So, and this we could do with SARCs because the PFPE contains fluorine, the other units do not. So you have a large scattering length density contrast between the backbone and these units. And so we did our SARCs measurements and we obtained intensity versus angle or Q. And we found out that polymers which contained I units, they had only from the Q position, we found out what is the length of the I unit, we found out the length of the M unit, because you see you have this reciprocal relation between the Q and the D, which is the distance. And not only that, if you look very closely, you will see that the polymer which has the highest mechanical strength also has the smallest full width half maximum, whereas the polymer which has the weakest mechanical strength also has a broad full width half maximum. So the SAS measurements were telling us that the polymer which had the highest mechanical strength was less dynamic than the polymer which was the other polymer. So this also gave us information about the dynamicity of the polymer coatings. And now we bring these polymer coatings to a test. So we put them in real cells. So what we did was we prepared a cell where there was lithium metal on one end, and we used copper because copper would give a good contrast if lithium is deposited on it. On the copper, we deposited our polymer layer, and we had 5 different polymer coatings. So we had 5 different cells with different coatings, and we also used a reference cell where we had no coating. We did lithium plating and stripping and looked at the coulombic efficiency initial cycle and after several cycles. So you can see from this graph here that if you have no coating on the copper, you have less coulombic efficiency. If you have a coating which has a very high mechanical strength, you again have a low coulombic efficiency. But if you have coatings of a particular ratio, so in this case, 1 unit of M, 1 part N units, 3 parts I units, this gives you the best coulombic efficiency. Also the other, which have less amounts of N units, they also give you a good coulombic efficiency. This means if the mechanical strength is low, the dynamicity is more. This is better for the cell performance and better for the coulombic efficiency. So far, so good. What about lithium dendrites? So for this, we looked at SEM images. So these 3 are the top view images of the copper after having washed the copper after cycling. So we've now removed the polymer and we look at the top. And you can see without a coating, you have lithium dendrites formation. If you use the coating called M1I0, so the coating with the highest mechanical strength, the lithium dendrites reduce in number, but you still have dendrites. But if you use the coating which is viscoelastic, which is less amount of M units, so mechanically weak and conformal, this gives you a very good lithium deposition, which is smooth and homogeneous. We, of course, wanted to look at the polymer coating as well. So we also looked at electrodes which still had polymer coating, and we tilted it so that we could see it better. And you can see that for the coating M1I3, which had a good smooth lithium deposition morphology, the polymer coating is still intact. It is still covering your copper. But for the mechanically strong polymer M1I0, the coating is not intact. So there are some regions where the coating remains, but there are some regions where the coating is ruptured and the lithium deposits are now, lithium dendrites are now coming out of these areas. So in the end, I think I'm almost end of my time. So in the end, I would like to say that we are able to use different neutron methods to look at different aspects of batteries and answer key questions on what really is happening when you change the components, how is the aging happening, and how we can use different techniques and find out what is the reason for the improvements in the batteries. So with this, I come to the last slide. So these are, so I acknowledge all the co-authors because these works were done in cooperation with a lot of universities and research institutions and companies. I thank the funding agencies which provided funding for carrying out these projects. And I thank you all for listening, and I'm open to questions.
Thank you, Dr. Neumann, for a very positive presentation on the future of energy storage. Any question? Is a question there?
The interesting talk. I just want to find out, I didn't really capture the purpose of the solid electrolyte interface. However, that was not really my question, but maybe if you can clarify that. My question is, since while you are dealing with nanostructures, in optimizing the production of them such that you have got interlinked pores for your electrolytes to pass through, do you have any particular model, or you just do it at random?
So I did not. So I understood the first part of your question. You wanted to have more information about the SEI, but the second part of the question was not so clear to me. Could you repeat that, please?
Yes, I just wanted to find out. I think these nanoparticles, you are actually synthesizing them. So I want to understand if do you just do it at random, or do you have a guiding computational model that you use before you can synthesize them?
So are you, do you mean the nanoparticles which we use for silicon, graphite composites?
Yes.
Okay, so in this case, we get these nanoparticles from the company Wacker, and then in our chemistry department, we mix the silicon nanoparticles together with the graphite, and we prepare the composites. We prefer the nanoparticles of silicon compared to microparticles because we feel this would give a better performance depending on the surface area. Is that answering your question?
Yes, I think you have answered my question. But then, how do you also confirm if they are actually if the pores are actually interlinked for the sake of the electrolyte movement?
Okay, so the thing is that if you have these, if you have such a structure with pores, if the pores are closed, if they are not accessible to any electrolytes, in that case, you would see a SANS peak coming for the protonated electrolyte as well as the deuterated electrolyte, because it doesn't make a difference on how you, on what is the, so in SANS, you get a contrast when you have a difference between the pore and the matrix, right? And in the case, and these SEI products, they have the same scattering length density as the electrolyte itself. So this we use in order to understand whether the pores are filled or the pores are empty. So in this particular case, because we did not see any SANS peak for the protonated electrolyte case, but only for the deuterated electrolyte case, this meant that there was the SEI there, which was there was electrolyte which was going through within the pores and was forming at the interface between the electrolyte and the solid.
Any more question?
Good day. I'd just like to ask, with regards to your doping of silicon on the anode, was it a surface effect or was the bulk also affected? Or, you know, what did it happen? You know, only on the surface, or you know, did the silicon actually reach the bulk of the material?
Mm-hmm. So
I think that's the first part of my question. Yeah.
Mm-hmm. So in this case, when we were doing these type of experiments, where we were mixing silicon with graphite, these mixtures were really good and homogeneous and going through really homogeneous within the bulk. Because you look here, if the mixture of silicon and graphite was only at the surface and not in the bulk, you would not get a good lithium content after fully charging the cell. So in this case, it was really, completely mixed in the complete silicon graphite composite. But I showed you another example here. We were trying to add silicon in titanium oxide nanotubes, and this we were trying to do with chemical vapor deposition. So we did not know if by chemical vapor deposition we are now able to coat the complete nanotube or just the surface. And it turned out, after doing the neutron grazing incident small angle scattering measurements, where we were probing the depth, we found out that the silicon which we deposited with CVD was just on the surface and not in the bulk.
Okay, then the second part of the question, when you looked at the formation of dendrites, did you consider the chemical composition of your anodes? Because if you look at maybe something like ICP-MS, the composition of your material to a large extent, tends to determine whether or not you get dendrites. So, for instance, if you have X amount of copper, especially in your material, you then be more likely to form dendrites. So, did you actually look at the chemical composition of your anodes at some stage?
Mm-hmm. So what we did in this example was we used the exact same amount of the same type of copper, and so we had cleaned. We took total 6 cells, and we used exactly the same type of copper in each of these 6 cells. The copper which was cleaned and prepared the same way from the same batch. And then we coated it with these different polymer layers, so that there is a common copper on which we deposit these different polymer layers. So the batch is not different, the cleaning is not different, it's the same. And yeah, the idea was just to see how the lithium would deposit on the copper. So we just did one example. So on one batch of copper, we did not try different batches of copper. So we had a uniform copper from same source, same cleaning for all these experiments. But you're right.
Yeah.
If you're using, it's like, it is true, as you can see here, that you need to have a very homogeneous anode, because if you have a not homogeneous anode and you have impurities, these or cracks, these or grain boundaries, these lead to hotspots, and these hotspots are the ones which eventually lead to dendrites which come out of the anode. So the anode quality composition is really important. That's true.
We are running out of time. I've got one last question. Dr. Nelman, I noticed you are part of the data evaluation group.
Mm-hmm.
Can you tell us in summary what the data evaluation group does on neutron facilities?
Okay, so this is a new group which we have formed. We have the experience now that there are several users who come, who do the experiments here, but because they are not able, so the data evaluation is sometimes not so easy for different neutron instruments. So you have to do data reduction, you have to do the data analysis, and this is something which is difficult for some people, especially the PhD students who are new. And sometimes it so happens that the PhD students find it difficult to evaluate the data. So they don't evaluate the neutron data, they finish their PhD and go, and the neutron data is left unevaluated. So to avoid these things, we started a new group with 3 scientists which have expertise in different instruments and different data evaluation softwares. And we offer this our help in data evaluation in exchange for co-authorship, of course. And the hope is to get more publications for our institute in this way and to encourage more new users to apply for Beamtime, because they know they will have someone to help them with the data evaluation after they do the experiment.
Yeah, thank you. Let's thank again the presenter. We are going to have a short break of 20 minutes. We are pressed with time, so not sure what time we should be back here. Just check. Supposed to start again at 55 past 6, no, past 3. I think we should have a short break for 10 minutes. Thank you.
Ich glaube, die sind irgendwie in die Google.
Good afternoon, Martin.
Yes, hi. Can you hear me? Hello, can you hear me?
I can hear you from Germany.
Hello.
Yes, hello. Cool.
He's talking, but I can't hear him.
You can,
hello.
Yes, can you hear me? I should be, I can see that my microphone is working.
Hi, Martin.
Yes.
Can you try again, please?
Yes, I'm here.
Yeah,
Yes,
yeah. There you are. Then you can beam your presentation. Let's see if it's okay.
Okay.
Don't forget your camera to switch it on.
Yes, I share my screen first. Can you see it?
Oh, there we are. Thanks very much.
So now I'm presenter mode and I will try to put on.
Just don't forget to share the screen.
I just did it. I just will put on the camera now.
Oh no, okay, no, it's visible. Thank you.
Yeah. Where is this? Where is my? So now you can see us, I hope, and I will share it. So my screen is still shared, I think, and I will go to the presenter mode now. Is it okay for you? Hello?
See you just now. We're going for a tea break.
Yes. Do you see my presentation is okay?
I see the presentation, Martin, and it looks okay.
Okay, good. Then we are going for a short coffee and we are back at 35. That's the schedule, no? Eigentlich ist es ja cool, oder?
Always something that müssen wir das und das machen.
so richtig, das ist ganz
Hi. Can we start now?
Yes, we are here. Do you hear me?
I think we are ready for the next speakers.
Okay.
My name is SA-SENS2022. I'm an instrument scientist with the neutron diffraction section at NEXA. I'll be chairing the next two sessions on magnetism. So our first speaker is Dr. Martin Boehm. He is the head of spectroscopy at ILL in France. So this guy, Dr. Boehm, has over 20 years of experience in the neutron scattering field. He started as an instrument scientist at ILL. He is very involved in studies of dynamics in quantum magnetic and correlated electronic systems. He is also active in applications of machine learning to inelastic neutron scattering with the aim of accelerating the data acquisition and interpretation. So his talk this afternoon is on magnetic order and disorder probed by neutron scattering. Dr. Martin, you can go ahead with your presentation.
Okay, thank you very much for this nice introduction. I have to correct it immediately. It's not only myself. We decided to share this talk. So it's also Thomas Serbeck, a colleague of mine who's here beside me, because we thought that it's also interesting to give not only diffraction inelastic, but also a bit of information on reflectometry, on magnetic reflectometry, and maybe some kind of applied aspects of magnetism with neutrons. So our hello goes to South Africa. I hope you have a better weather than we are. We are in autumn. I guess you're at the nice springtime just right now. So we would like to share that with you, but it's fine like that. So let's start with the presentation. So very quick overview. So I apologize immediately if it will be too general, but I thought we were asked about magnetism and neutrons, so I thought that not everybody is expert in magnetism, so I give a brief introduction. I show about the LL, but I guess you had some transparencies already this morning by Mark Johnson, and then I'll show quick, very general things about neutrons and magnetism, and then I jump to more specific examples in ordered magnetic systems, and then I switch over, and Thomas will continue with polarized neutron reflectometry. So very quickly about the LL, I suppose, I don't know, that Mark maybe have presented it already. So the LL is in Grenoble in the heart of the French Alps. And you can see what, I hope you can see my pointer here. What I show you here is what we call the polygon scientifique. This is an island between two rivers in the French Alps here in Grenoble. And this whole area here, why I'm showing you this today, is because this whole area is more or less has developed by the initiative of one person here, among others, of course, but one key driver during and after the Second World War, which was Louis Néel. He got a Nobel Prize for his work to antiferromagnetism, and that's why I present Louis Néel especially today. So he was director at the CA, which is here behind. Afterwards, director of a magnetism laboratory, which is nowadays called after him. It's the Louis Néel Laboratory. And also he was the driving force for the LL, which is here. So our office is just here where you see this red flash. We're sitting here right now. And beside us, again, in the 80s, there was the ESRF, a synchrotron radiation source, which was also initiated by Louis Néel. So if you're not familiar with magnetism, Louis Néel is very important because he predicted a particular magnetic structure in condensed matter systems. So he claimed that there is a doubling, or there's a possibility of a specific magnetic structure where the atomic spins point antiparallel in the unit cell. So example is here, mangan oxide, when the chemical unit cell is given just by these neighboring atoms, but you see that the translation symmetry is broken, and you see that the next neighbor has an antiferromagnetic spin with respect to this one. So we are doubling the unit cell, the magnetic unit cell, and this can be directly probed with neutron scattering. So below you see here, this is the nuclear structure, which was measured on powder. This is a powder nuclear structure of manganese oxide at 120 Kelvin. And if you cool down and you go below the ordering temperature, which is now called Néel temperature after Louis Néel, you see that there are additional peak arises, and this additional peak are indicator of this superstructure. So you're doubling the cell. So in reciprocal space, you have more magnetic peaks in between the nuclear peaks. So this second work, this is a pioneering work in neutron diffraction, magnetic neutron diffraction, and the Nobel Prize also went to Clifford Scholl at that time and Bertram Brockhaus for the development of the neutron diffraction technique. So nowadays, of course, the magnetically ordered systems play still an essential role in our business here with neutrons. So I would love to give you more dedicated examples, but I think it will be not. Won't have time to that. I just mention one which is very topical, fancy topic still. This is multiferroic systems where you can see that the magnetic order is coupled to the polarization, electric polarization, and you can switch in between them. But what I wanted to say, and here I follow a bit an article which I found in Nature in 2016, because I found this very nice. We are not only studying ordered magnetic systems, but there were major two discoveries, I would say, which changed the field a bit and went more into the direction of correlated electron systems and quantum magnetism. So the first impact was in the 80s with the discovery of high DC superconductors. And I suppose that most of you have already seen this phase diagram where we show temperature against the doping of let's say, a family of high DC superconductors. You see that it's first antiferromagnetic, but with higher doping, you come into the superconducting dome. And around the superconducting domes, with the time, we discovered there are a lot of competing electronic phases. And the superconductivity, in fact, is not a conventional superconductivity. It means that the Cooper pairs do not bear with a phonon intermediate force, but they are there is supposed to be that there's some kind of magnetic interaction involved in that. And that's where the inelastic neutron scattering comes into, because these magnetic resonances were found very early, and they still play an essential role in understanding what kind of mechanism might be behind the superconductors. So the high DC superconductors, they are 2D metal, 2D strange, let's say mod insulators, but 2D compounds, which triggered the whole field more or less into the low dimensional and low spin applications, and this goes more and more into this quantum world. But there is a second influence or second impact in this condensed matter physics. It's physics field, and this is the topological states of matter. So in 2016, Haldane, Kosterlitz, and Toules, they got Nobel Prize for their work, where they more or less apply topological systems into this quantum world and map this kind of physics onto quantum systems, many-body systems. And especially we here at the ELL are very proud, because Duncan Haldane was a postdoc in the 80s, so part of his work was developed here at in Grenoble. And again, this kind of systems develop more, or are developed for or are very showcases more or less are low dimensional spin systems. And this again brings us into the field of quantum magnetism, where we try to understand what is going to happen in at low dimensional, so 2D or 1D systems, especially when the spin number is low, and in addition when there is maybe some kind of geometric frustration present. All of these three ingredients provoke what we call quantum magnetism, and a very interesting applications behind, studies behind. So all in all, the activity, when I say magnetism, so it's not only the ordered magnetism, but this whole list which we see here, as I said, also about correlated systems and quantum magnetic systems. So this whole activity is about 30% of the LL, which you can see here. So it's a very important field in neutron scattering. So this is now very quickly. I don't want to bore you with some general things, but what I want to mention is that when we are talking about in magnetism, what we really look on are the electrons, of course. So the unpaired electrons in any kind of compound. And you can imagine that they create some magnetization, and the neutron probes this kind of magnetization inside your compound via dipole interaction. So this gives rise, so the probability that this interaction takes place, or the probability that you scatter in a certain angle with a certain energy transfer, this is proportional what we call the S of Q and omega, so the scattering function. And the scattering function, as I wrote it here, is some kind of expectation value of this magnetization, but in a reciprocal space. So we go from the real space into reciprocal space, and we go from the time domain. So the time dependence of the fluctuate, or the time dependence of the spin correlation transforms into an energy channel. So this S of Q and omega gives you a complete microscopic description of your system, and the intensity which we measure in the detector is direct proportional to the scattering function. That's important. So that's what I again wrote here. So the scattering function, so that's what we really measure. You can say it's proportional to the intensity which we measure. Is more or less the Fourier transform of the time-dependent pair correlation function, or spin correlation function. And the last thing, a detail, but an important detail, is that there is a polarization factor inside. Due to this dipole interaction, this character, we see only the projection of the magnetization onto this Q vector in reciprocal space. So we only see this red contribution. So if you get bored by these equations, I put in a small cartoon, a short cartoon about the interaction between neutrons and the spin system, a two-dimensional spin system. You can see that when the neutron has an interaction with the system, you create some correlated movements, which we call spin wave excitations, spin wave excitation. And this spin wave excitation, of course, can be calculated, and we can calculate the intensity and compare with the experiment. So that's what I want to show here. It's what I've mentioned is, of course, we have a momentum space. So we have a three-dimensional reciprocal space, a three-dimensional. So we have this Q with this 3-dimension, and we add a fourth dimension as the energy transfer. So what we are looking at is intensity, the intensity distribution in a 4-dimensional space, which is, of course, a difficult issue. So we try sometimes to plot and find tools which can visualize a 4-dimensional space. And here's something which is stolen from in fact, from medical application, where which is used for tomography in medical imaging. And we applied it to a simple spin wave calculation. It's not that simple, sorry. It's a Skyrmian lattice excitation. And you can see that we kept one of the dimensions fixed. So one of the reciprocal directions are fixed, but you can see still 2 dimensions in reciprocal space. And the axis perpendicular is the energy transfer. So we can make cuts through this kind of excitations. And I show you this because in the examples afterwards, I will show you various cuts through this 4-dimensional intensity surface, which are either 2D, or sometimes I just show a plot which is taken by a 1D scan. So I just vary, for example, energy, and you can see the intensity distribution along one axis. So of course, this 4-dimensional space can be reduced when we're using powders. The powders average out let's say the directional information, and only the magnitude of the Q vector is conserved. So this gives spectra in 2D, so the momentum transfer via the energy. And of course, if we ignore that there's an energy transfer, then we come just to the diffraction case. So we just explore the scattering in the reciprocal space. So compared to other techniques, I believe there are 2 major issues for magnetism which makes neutrons very, very valid. The first one, of course, are the dynamic conditions. So because, I mean, you all know this with the de Broglie correlation, is that the momentum of the neutron is related to the wavelength, so that the wave vector, which already gives you some information in diffraction. So this matches the speed, and the mass matches perfectly the wavelengths which we need, about angstrom size, which gives very good diffraction signals, or larger length scales afterwards, where you go to nanoscale, structures. But also, and this is mentioned as well, due to this I think more or less by chance, nature gave the neutron a mass, which gives it a kinetic energy, which is perfect to probe magnetic excitation. So we get with this kind of mass and speed, we get energy scales of milli-electron volts, maybe a bit longer with cold neutrons. And this milli-electron volts is perfectly for the domain for spin dynamics. So we are in the picosecond region here. So that's where we do the classical spectroscopy with time of light and triple axis spectrometers. And then when we go to longer time scales, we go to back scattering and spin echo. So of course there are other techniques around, but they don't have this nice Q dependence as we have. The second thing, which is really important, is of course the kind of interaction. And I mentioned before that there's a dipole-dipole interaction because the neutrons has a magnetic dipole moment, and you can use that. I will mention that in context with the polarization factor. You can write this correlation function in a bit different way in difference to just mention the projection, which you can write out in a full term, and you just consider this component behind, and this is now the spin dependent correlation function. You can really, dependent on the incoming and outgoing spin configuration of the neutron, you can probe specific elements with polarized neutrons. So there's a film which a colleague of us, Navid Queshri, made for a technique which is Cryobat. I don't know if this was a bit fast, if I can play that again. You can see there is incoming neutron spin, and you can, with magnetic fields, you can tune that, the incoming direction. There's an interaction at the sample, and afterwards you analyze the spin state again into the detector. And this additional analysis of the spin state gives you very precise information about the magnetic system. We have another one now coming online. This is a publicity for something which we call BASTIS-3. This is something which comes on the powder spectrometer at ERL. So this is a so-called wide angle polarization analysis system, which gives access to a complete scattering plane for certain energy transfers, but in polarization state. So now I'm coming, don't know about the timing. I think I will focus on one example and hand over then afterwards to Thomas, otherwise it will not be sufficient time. So I wanted in my example, especially highlight complementarity between neutron techniques, but also with other techniques, not only with neutrons, but for example, soft RPS. What is essential in all our systems nowadays is, of course, that we have very good models, and in order to calculate our spectra and compare that with the experimental results. And new experimental possibilities, of course, is always something that is driving our field, and I just mentioned BASTIS before. So I will, so BASTIS, just to show you what we can do, as I mentioned, this is something where we can scatter out in a scattering plane. You can see that here. This is the coil arrangement. So the incoming neutrons are here. They hit the neutrons, and afterwards they scatter into this wide angle detector system, which we call flat cone. And this is how it looks like for this is a cut at a constant energy, and you see two different directions in the Q space. And you can see a one-dimensional magnetic system. It's only dispersive, which you can't see perpendicular to the scattering plane here, but it's constant in the other direction because there is no magnetic interaction. So these types here are, there are different kind of signals. It's not only magnetic signals, but there are also phonon scattering here, which you can see here. And this you can very nicely separate out with this XYZ polarization analysis. And you can separate now only the nuclear contribution, which comes from a phonon scattering, and you can hear different channels for the magnetic signals. So now one example which I've chosen, which gives you some, I hope, give us some kind of overview about diffraction and about spectroscopy, what we can do nowadays. It's a Skyrmian lattice in a chiral magnet. The compound is very well known. It's manganese silicide. And I show here the phase diagram. So you see the magnetic field is a function of temperature. And you can see when the zero field, when you're in zero field, the magnetic structure is helical. So it means that the spins turn around propagation direction perpendicular to it, and they form a helix in a specific direction. When you put on a small field, we go from a helical into conical states. So we get an additional ferromagnetic component into this helix, which gives this conical state. But as you can see here, what we are interested in is this small pocket here. This was called the A phase. And in the meanwhile, it's clear that this is a so-called Skyrmion phase, where in conjunction with the magnetic field and the specific interaction, which is called Dzerzhinsky-Maria interaction, you can form a very specific topological object, which is called the Skyrmion, and you can see a sketch in a real space, in a two-dimensional real space. When you try to probe that with neutrons, you will realize that it's a so-called multi-K structure. So you need 3 different K components in order to reconstruct the structure. And these 3 components you can see directly with a small angle neutron scattering. So you send in, and the incoming beam is here. You apply the field in the same direction as the incoming beam. That's important, and you will scatter out due to this Skyrmion lattice into, in this case, 6 different peaks, because you have this threefold multi-K structure, but you also have the minus K, so you end up with 6 spots in reciprocal space. So this top one is the conical phase. You can see there's a different symmetry here, but when you come into this A phase, you have this hexagonal symmetry. There's a direct proof of this Skyrmion lattice. And the very fancy thing about these Skyrmions is that they always stay perpendicular to the field. And that's what you see in the lowest one. So they turned the crystal around the arbitrary angle, and you see that you still keep the same diffraction pattern, which means that this magnetic Skyrmion structure is completely decoupled from the underlying nuclear lattice, which is a very cool issue. So we can go further than that and look what kind of magnetic excitation you might expect with that. And here, so you have, again, you see here the Skyrmion lattice in a 2D plot. We apply a magnetic field here. So you create this local Skyrmions, which you can take as a local magnetic field here. And the question is now, what kind of excitation would you expect? So how do these spin wave propagate in the presence of this topological objects? And here is what you can see here. This is when a free, let's say, free propagating spin wave comes in the proximity of this Skyrmion lattice. You can see that the local frame, the local coordination frame, change on the surface, or on the circular surface of this Skyrmion. And when you propagate around here, you can see that the magnon acquires an additional phase, a Berry phase, which is a direct proof for this topological object. So here you can, the plot what you show here is shown where you have a topological density around this object. And our colleagues in Munich calculated the kind of excitation which you might expect, and they realized that when a magnon comes close to this Skyrmion, that they have what they called confined levels, Landau levels, similar to electrons in the magnetic field. And with inelastic spectroscopy, we could directly prove this kind of excitations. They are the distance between these surfaces is very small. It's of the order of 10 microelectron volt, which is usually below the energy resolution which we can have. So what you can see here, this is a standard triple axis spectrometer. And as I said before, we just have a one-dimensional scan. So we plot intensity as a function of the energy transfer. You can see when you go through, we see broad excitation spectra, which corresponds to, this is just the calculations, which corresponds to specific directions here in energy. And you can see that we integrate over a lot of different levels. Nevertheless, some of these levels have some kind of polarization. So, and you can separate that with plus, minus, minus, plus polarization. So they are differentiated here in blue and in red. And this is exactly what we also can reproduce here in the experiment. You see here one of the channels of the spin flip channels, and here's the opposite spin flip channel, and we can exactly reproduce with the experiment the theoretical predictions. But we can, with a different technique, with the Mieze technique, which is used in Munich, we can go to much higher resolution. It's a specific spin echo technique, which has a much better energy resolution. And here we can really resolve the different energy levels coming from this, which corresponds to this kind of levels here. In total, there are 14 prediction levels, and some of them can be reproduced by the experiment. So I will skip the other examples because I think I take too much time, and I will hand over now to Thomas for the reflectometry part.
Yes. Hello, everyone. And well, thank you. And thank you, Martin, for giving this opportunity to talk a little about reflectometry. So my name is Thomas Arvik. I'm an instrument scientist here at the ILL of the instrument D17 here right in the middle. And I'm within the large-scale structures group where these reflectometers are located. Here at the ILL, we have 3 reflectometers. And basically, in terms of the science areas that we cover on those 3 reflectometers, you find everything from soft matter over chemistry, biology, biophysics. We also investigate batteries and in the energy sector. But also what I do mainly is magnetism, which is what I'm going to talk here about today. So as I said, I'm coming from the large-scale structures group, and I'm investigating nanoscience. So how do I mean that? What is nanomaterials, and why do we talk about large scale? Well, everything you heard in Martin's presentation was basically on the atomic scale, so spin wave excitations and atomic scale. So what we do not do is diffraction or nuclear physics. Everything nanomaterials and large scale for me is everything thin films, nanoparticles, nanostructures, or domains. And luckily for me, this is also in the realm of applications of new technologies. And whenever I'm writing a new grant, everybody knows that you have to kind of say, oh, this will be the applied new technology in the next new quantum computer. And in fact, this is what we're doing. And so today I just thought I'd give you 2 examples of what are real applications of polarized neutron reflectometry on today's spintronics or new computing device systems. So the first one is here. I'm sure you have heard about a magnetic tunnel junction, which is basically implemented in this new, very fast, and at the moment still very costly chip of the MRAM, magnetic random access memory chip, which is basically consisting of 2 ferromagnetic layers separated by a thin tunnel barrier. And by the orientation of these 2 ferromagnetic layers, you can choose your information state between 0 and 1. And so that can be implemented in magnetic retards or in this Everspin MRAM chip. Now, the basic ingredients for such a magnetic tunnel device is a magnetic layer, which in this case is cobalt iron boron, and a thin insulating layer, which is in this case MgO. These layers have to be very thin, which means there is a high requirement on the interfacial structure, which is shown here with TEM on the right. But also in terms of the quality, we have to do something to get the structure even better, to have it crystallized and really sharp interfaces. And that is typically done industrially by annealing. And I think everybody of us knows that if you heat up things too much, they don't like it. And so as soon as we go to higher annealing temperatures, there's a deterioration that sets in, which the industry is currently struggling with and trying to find ways out of it. And so here from our neighbors at Spintech, from Bernardini, who is one of the spearheadings of this magnetic tunnel junction MRAM assembly, they're trying to play around with diffusion barriers, and we did reflectometry on some of his samples to see what's happening. So what are we looking at here? Well, this is basically the end result of a polarized neutron reflectometry experiment. And I'm showing here an experiment done with neutrons and with X-rays. So one of them is on the upper panels, you see neutron reflectometry. On the lower panel, you see X-ray reflectometry. We have here a couple of lines, and what these lines are basically a description of your sample as a function of depth. You start at the substrate, then you have a buffer layer, then there's a tantalum conducting layer, then we have one MgO barrier, we have a cobalt iron boron layer, we have another tantalum layer, platinum layer for the upper contact. And that is basically a description of how your sample looks as a function of depth. in this case, just the nuclear chemical information, which you can also obtain by X-rays, but where the neutrons measure the nuclear contrast, the X-rays measure the electronic contrast. So we have two contrasts mixed in one system. And what the neutrons give you in addition is the magnetic information. So what we're looking at here in the center on this part here is the magnetism inside the sample, and in this case inside this cobalt iron boron layer, which was 18 angstrom 6, so 1.8 nanometer. And we start to anneal down here as a function of temperature. You see red is hot, blue is cold. We have first a nicely defined magnetism inside this layer, and as soon as we start annealing, this whole peak seems to shift towards the MgO barrier, and then it drops off. And this is basically the problem, and why you can actually see that is here as well. You have diffusion of these kind of elements, tantalum and platinum, inside this layer, which causes this disturbance here. And you can also see that in the X-rays. As soon as the tantalum comes in, which is a high electron material, the magnetism drops away. So what is wanted is this shift towards the interface that is good for MTJs or magnetic tunnel junctions. What is not good is when this tantalum comes in. And so what they tried is to put a tungsten diffusion barrier in there. And here actually, as a function of temperature, I don't have to explain much, because you can see there is not much happening. It shifted towards the interface, but it stayed where it is in its full magnetization. And I should say this is magnetization in absolute units, so you don't have to do any fancy calculation of like with squid or fitting of response curves or so. This is directly a measure of the absolute magnetization in the cobalt iron boron. And so we can separate all these effects. This is just a summary slide, which is basically showing the diffusion of the elements inside the layer. So the nuclear part, where we see iron and boron walk into the tantalum layer, the platinum walks into the tantalum layer. They create this large enhancement in nuclear contrast inside the tantalum layer. But iron and platinum don't really like each other, and so they kind of stay apart, which is basically leading to an agglomeration of them and stealing the magnetism of one layer, which is why this magnetization shifts towards the interface. And as soon as the tantalum breaks in, which is shown here, we have this drop. So for the second example, actually, I come to a multi-layer system, and Martin talked a lot about antiferromagnets on the atomic scale. And unfortunately, in magnetism, there are not many techniques that can really see antiferromagnetism on the nanometer scale. And neutrons are really wonderful for that. This is an example which was first published here in the Science article, coming back to the giant magnetoresistance effect, which was a Nobel Prize in 2007. And here it is shown for the first time in an all-oxide magnetic multilayer, where you can see when you cycle through the magnetic hysteresis loop, you have a saturated state where all of these layers are pointing in one direction. You go to the remnant state and you have got this antiferromagnetic alignment due to the RKKY-like exchange coupling between these different LCMO layers on the atomic scale. So this is a large scale antiferromagnet. It's not an atomic scale antiferromagnet. And we can use neutron reflectometry to really look into that. So first, what we have here now, this is the reflectometry data, which we fit by a model. And this is basically the same that we saw before. It's the description of your sample as a function of depth. But what we see here in red is the magnetic signal, where all the magnetizations point one way, which is here our ferromagnetic state. So if we then go to the remnant state, it is like we would expect. We have a pure antiferromagnetic state. We can immediately see that from the data, because you can see here from this Bragg diffraction peak, it shifted to the half order, which means a doubling of the unit cell, as Martin has explained. And basically that immediately tells us there's an antiferromagnetic component here. Now we have antiferromagnetic alignment. So if we now take a look back at this hysteresis curve here, the intermediate plateaus here, and actually this slide shows something else, I'm sorry. You see in the remnant state, the hysteresis loops don't really meet. So there's a question, what is here? Is that an artifact or something? But we did an experiment here as well. And what we could see is actually that there is a difference between those two remnant states, and that is within the antiferromagnetic alignment. I know it's not very obvious, but if you stare at those two blocks long enough, you will see that the order has reversed. Here, the first layer points to the right. Here, the first layer points to the left, and afterwards antiferromagnetic. Otherwise, these two are exactly the same. It's just that they are 180-degree reversed antiferromagnetic states. And to my knowledge, there is no other techniques that can distinguish such a case, which has an average magnetization of 0, and it's just which layer order is where inside of your sample. Polarized neutron reflectometry gives you direct access to that. Now, the last thing that I wanted to point out is what happens on this intermediate states here. And that was the interesting part. So when we do the measurement on these intermediate states here and we look at the profile, what we simulate is you see that one layer either near the substrate for the increasing loop branch or near the surface for the decreasing loop branch nucleates into a ferromagnetic state. And then when you increase the field, you basically have this like a soliton propagation throughout the film. So again, polarized neutron reflectometry can immediately distinguish which layer has switched where in the system, depending on which loop branch you are. And that basically can give you a nice layer resolved magnetization of the system. And that already brings me to my conclusion. The only point that I really wanted to make here is, because I'm doing it every day and it gets me excited every day, is that neutron reflectometry is really a perfect tool to study magnetism of thin films, nanometer structures, which have real world applied chances. So not all of this will at some point come to applied technologies, but we still keep our fingers crossed for our next grant application. So with that, thank you very much, Martin. I'm handing back over to you. I'm scooting to the side. I think we're done.
Yes.
Yes.
Hello.
So do you have any questions?
Switch to the last one.
Thank you so much. Do we have any questions? I want to know, do you perform this experiment in sample environments, and are they ferromagnetics or anti?
Maybe I start answering the question. Yes, of course. I mean, all these systems happen at very low temperatures. Sorry, can you hear me?
Martin?
So the magnetic order, of course, happens at low temperatures, especially for the quantum systems. They are very low temperatures. So we rather routinely have now dilution fridges, which cools down the samples to 50 millikelvin. And these can go inside magnets. So we have magnets which can go up vertical magnets with a magnetic field perpendicular to the scattering plane, which goes up to 15 Tesla. But we also have horizontal magnets. So we have a broad spectrum, let's say, of external parameters which you can apply. So we have temperature, low temperature, we have field, we have also pressure cells. And we can combine all these together, usually in order to create extreme conditions and to test the different cases which we need.
Any questions before we close the floor? Thank you so much for your presentation.
Okay.
Well, thank you for having us.
We can go to the next speaker. Is Dr. Nikolay Kajilov.
Hi, good afternoon. Do you hear me?
Yes, Dr. Nikolay Kajilov is a researcher at the Institute of Applied Materials in Germany. He also started as a beamline scientist for imaging instruments. He is a member of the Joint Research Unit Nickel Matters, supporting construction and operation of new neutron imaging instruments next at ILL. He has vast experience in the development of methods using different contrast mechanisms, such as in diffraction imaging and visualization of magnetic fields by polarized neutrons. You can go ahead with your presentation. Thank you.
Okay, thank you for the invitation. I'm really very glad to present my talk in your workshop. So first I should export my screen. Just tell me if you see it. Do you see my presentation?
Yes, thank you.
Okay. So today I'll speak about neutron imaging. Neutron imaging is transmission neutron technique, where the neutrons are transmitting the sample, and we see just the transmission image, which is taken by a position-sensitive detector. So this is different than the scattering techniques, where we see the scattered neutrons. Here we see the transmitted neutrons. So, you know, it was already discussed quite broadly that the neutrons obey the particle wave duality. So we can represent the neutrons as a particle, which is a very common representation for imaging experiments, where we count actually neutrons which are transmitted through the sample and look for the attenuation of the beam. But the neutrons can be represented also as a wave. So we can assign a wavelength, we can assign kinetic momentum, kinetic energy, and then describe the neutrons. So they're very unique. This is unique radiation, which can be considered as propagating particles or propagating wave. And all these kinds of interaction with the matter, so we can have interaction which is based on only on absorption, but also we can have diffraction, which is related to the wave nature of the neutron. And all these types of interaction actually can be used for contrast for imaging. So even the feature of the neutron, that it has a magnetic moment, can be used also for imaging. And these are the so-known polarized neutrons, where the spin of the neutrons or the magnetic moment of the neutrons has some preferential orientation. This means that you can imagine that the neutrons are just a compass needle. So these are small magnets which have the same orientation. This means that when they pass through a magnetic field, they start to oscillate around the external magnetic field, and we can use the angle of the precession actually as a contrast mechanism, as it was done here, visualizing a permanent magnet. So you see in the middle the permanent magnet. So we just send the polarized neutrons, so the small magnets, through the magnetic field which is produced from this magnet. And what we see at the end is the oscillation angle of these small magnets, or the spin of the neutrons in the external magnetic field. And we can visualize this with a position-sensitive detector. So why neutrons? They have a magnetic moment. This magnetic moment, or the spin of the neutrons, interact with the magnetic fields, and we can depict actually the Larmor precession by using of position-sensitive detector or just standard imaging. So you can see here the experimental setup which we are using for imaging. We have neutrons. They can possess two spins, spin up and spin down, or two different orientation of their magnetic moment. We use a spin filter known as a spin polarizer, where we select only one orientation of the spin of the neutrons, and we send these magnetic moments through the magnetic field or through our magnetic sample. And after the interaction of the spin of the neutrons with the magnetic field, we have some spin rotation, or this precession, which is analyzed by spin analyzer, which transmits only the component in the direction of the orientation of the analyzer. So this means just a part of the original orientation of the spin. So this means that the intensity is really proportional to the angle of spin rotation. So here you can see again the representation, how the magnetic moment or the spin of the neutrons is oscillating around the external magnetic field. So this is known as a precession. And the speed of precession is proportional to the external magnetic field. So here, this is how the contrast is formed after the transmission of the polarized neutrons through the magnetic field. We have, this is the attenuation contrast, so the neutrons are also absorbed or attenuated from the sample. But we have another term which describes the spin rotation. So this means that the intensity is proportional to the cosinus of the spin rotation angle. So this is what we see here in our picture. So we have rotation actually from 0 degrees to 90 degrees. And when we have 90 degrees rotation, this means that we have the minimum intensity. And if we have 0 and multiple times, let's say 2 pi rotation, then it is a maximum. And due to the fact that the magnetic field of the permanent magnet decays with the distance, so we have multiple times rotations passing the 0 position and the pi position. So this means that this annual, let's say, structure of the picture, what we see here is the minima and maxima which describe the rotation of the magnetic field, of the magnetic moment of the neutron in the external magnetic field. So this was actually developed in our group in Berlin using the neutron imaging instrument here, it's 14 years ago. And this nice result was published actually in Nature Physics in 2008, and we are very proud of this. Of course, this is very nice technique for visualization of magnetic fields, and we use it also for visualizing different magnetic fields produced from dipole magnets. Here you can see here the multiple rotation of the neutron spin in the external magnetic field of these two dipole magnets. But the most interesting thing is that we can see the magnetic field here also in the aluminum slab, which we used as a spacer between these two magnets. So this means that we are able also to visualize magnetic fields in the inner of materials, which is really not possible by using of other techniques. So this is how this changes if we use non-dipole magnets. You can see that let's say the rotation of the spin or the field is not so symmetric as using well-defined dipole magnets. So this is just a qualitative type of information which we can get. And also I want to point out that what we see here is not the magnetic field. This is just the rotation of the spin of the neutron in the external magnetic field. So the way to reconstruct the magnetic field takes some more, let's say, efforts. So we went further, and when we get the information that we can see magnetic fields inside in bulk materials, we went to one very interesting example, and these are superconductors. In superconductors, we can pin or we can trap the magnetic field when they're in superconducting state. And when we increase the temperature, then passing through the critical temperature of superconductivity and going to higher temperatures, then this magnetic field, which was trapped inside in the superconductor volume, will be released. And we can observe this in one sample here. This is yttrium barium copper oxide, and you see that we trapped inside in the sample volume magnetic field. And when we're passing the temperature of about 90 Kelvin, this magnetic field is released, and you can see due to the temperature gradient, it is sticking here to this edge. I'll show you this nice movie again. So we increase the temperature, we trap the magnetic field inside due to the spin precession of the neutrons. We see this as a dark signal. And when increase the temperature, this magnetic field is released, and we see a small point here, which is due to the temperature gradient in this big, actually a big quarter of superconducting sample of about 4.5 centimeters and 1.2 centimeter thickness. We can go even further. and use a bulky sample of lead, which is polycrystalline. Lead has critical temperature for superconductivity of about 7.2 degrees Kelvin. So this means we can apply external magnetic field, cool down the sample with the field, in the field. And when it's superconductive, we switch off the magnetic field, outside external magnetic field, and we see that the field was trapped in the bulk of the superconducting lead cylinder. Then we started to increase the temperature, and just below the critical temperature for superconductivity, we see that probably due to some temperature gradients in the sample, we see only 2 points where the magnetic field was trapped. When we know that the magnetic field was, let's say, parallel to the axis of the cylinder, then we can rotate the cylinder around perpendicular axis and perform tomography experiment. So from this, we can reconstruct the magnetic field, the trapped magnetic field in 3D, and to know where it is located, which was also quite interesting. But the main question is, what is the strength of the magnetic field, or what is the quantitative value of the trapped magnetic field? So we applied some iterative method to get it out from these pictures, but it was still not a very major method. So it was not very well developed method. So we used the fact that the magnetic field, the trapped magnetic field, has one defined orientation. We used very homogeneous magnetic field which was perpendicular to the axis of rotation. This was the assumption to estimate this magnetic field. If we have some arbitrary field in all directions, so you know that magnetic field is a vector field, then how we can reconstruct this vector field from tomography experiment? For this, we need some special arrangement known as a polarimetric arrangement, where we have a analyzer. This means we can select the orientation of the spin of the neutrons in one certain direction, and then we have flippers. So if we imagine that we represent the orientation of the spin of the neutron after each step, so the analyzer is giving us one defined orientation. With the first flipper, we can turn the spin of the neutron in the forward direction. And with the second flipper, we can turn it in the plane, in the horizontal plane in a different angles. So after that, we can use the third flipper to set up again the orientation of the neutron after the interaction of the spin of the neutron in the plane. Then again to bring it upward and send it to the polarizer and analyze it. So it's a bit complicated, but we can actually perform experiments where we send the 3 different orthogonal directions of the spin orientations using this setup. So we can send, let's say, the X direction of the spin through the magnetic field and analyze how this X direction was influenced by the magnetic field, and what is the component after that in X, Y, Z directions by measuring actually the projection of the spin precession in the X, Y, and Z directions using the flipper. So this means that for one certain orientation of the spin, we perform 3 images where we detect the projections in X, Y, Z. So this means for one direction, we have 3 images. And if we send the 3 directions, this means that we need 9 images at the end. And from this, we can reconstruct actually the components or let's say the coordinates or the projections in the X, Y, Z directions for each vector, and reconstruct the vector direction for each point of our, let's say, magnetic field or of our sample. So this we used by using a known sample And you see that the simulations, just by knowing the geometry of this small solenoid, agrees very good with the measurement which we performed. So at the end, we used again superconductive lead sample. This was a quarter, and we were able to reconstruct the magnetic field of the trapped of the trapped vector field or of the trapped magnetic field, so we can reconstruct the vector field in 3D. So I think with this we could say that quantitative magnetic tomography is available, and I hope that it will be developed for the future in a much better way, that it is available, let's say, at the neutron tomography facilities over the world. So here, of course, if you have current flow, this produces also a magnetic field. If you have some frequent current sent through a conductor. And if you have a bulky conductor, then you can observe also a skin effect, where the current is squeezed to the surface of the bulky conductor when you increase the frequency of the current. So this we observed with the polarized neutron imaging. So we are sensitive also to current flow, and we can see here how the current density becomes very narrow, close to the surface of the conductor. This method can be used also for phase transitions from ferromagnetic to paramagnetic state. In the ferromagnetic state, we have a lot of magnetic domains inside in the sample, which depolarize the beam. So we send the beam with preferential orientation of the spin of the neutrons. After interaction of the magnetic domains, we have at the end some arbitrary orientation, which reduce actually the intensity when we go through the analyzer. By increasing the temperature of the sample, we go to the paramagnetic state, where the magnetization is no more there, and then we have actually preservation of the spin orientation of the neutrons after the interaction with the sample. So we have increased transmission. And if we plot the spin polarization for two different plots, you can see that you see immediately where this phase transition happens, at what temperature. So we can make some maps, temperature maps, for example, Curie temperature maps for such samples. And this was palladium nickel crystal, where the Curie temperature depends on the concentration of nickel. So we see some inhomogeneity of the nickel concentration in the sample by using of this method. So it's quite interesting. So the next method, which is sensitive to magnetic structure of the samples, this is a grating interferometry. We used some coherent neutron beam, which is prepared by a grating. So this means we use multiple line source, which produce some spatial coherence in the horizontal plane. And this spatial coherence can produce interference pattern by using a phase grating. So this phase grating is producing an interference pattern. And by scanning of the analyzer grating, we can get some signal from this interference pattern. So here, this is the phase grating. And at the end, we can analyze this interference pattern for each point of the sample. And for example, plot the amplitude of this interference pattern. When we have some, let's say, scattering in the sample, then this coherence is already distorted, and the amplitude of this interference pattern it becomes low or vanishes. So this means that we can detect areas of the sample where we have some scattering. So if we have magnetic domains in the sample, on the domain walls, we have something misorientation of the magnetic field or of the domains, and this produces multiple refraction, which actually is seen as a scattering This is area where the coherence of the beam is distorted and the amplitude is reduced. So this means if we can make a position-sensitive map of the amplitude with this grating interferometry, then we can see actually the domain walls very good. So this is iron silicon crystal, which was a model system with magnetic domains, as shown here. This is the absorption contrast radiography, just putting this small disc in the beam. And we barely can see it because it was 0.1 millimeter thick. So it is very thin foil, which can be actually resolved very well with dark field contrast or with the grating interferometry, and we see the domain walls very good. We can go further and perform tomography and see the 3D distribution of the domains or the 3D network of the domains in the sample, and make some quantification like how the domain width depends on the crystal thickness. So you see here that by decreasing the crystal thickness, we have different domain size. And here these points were provided by the tomography experiment, and the theory actually was confirmed with this experiment. So in addition to this, we can see also domain walls in electric steels, which are used for transformers. And if we put this electric steel in frequent magnetic field, these domains start to reorientate, usually in the standard electric steels, which increase the energy dissipation in form of heat. This means that if we have standard electric steel, due to the reorientation of the domains in frequent magnetic field, it produces a lot of heat. These are the old transformers. Nowadays, there is a method of laser pinning of the domains, so they cannot reorientate. But if we put them in frequent magnetic field, they change their size with the external magnetic field. This means that the energy dissipation is much lower as the full reorientation of the magnetic field. And this was observed with this method. So you can see here with the grating interferometry, we can see the domain structure, the domain walls in the electric steel. And if we put this electric steel in external magnetic field, then even though we can perform dynamic experiment where we see how the walls actually of these domains change at different, let's say, values of the external magnetic field. Okay, so you see quite exciting experiments, nice techniques. And but the time is limited, so I would like to stop here and thank you for your attention.
Thank you so much for that interesting presentation.
You're welcome.
Is there any questions?
Hi, Nikolai. Thank you very much for a very nice talk. My question is that this instrument, what's the future of this instrument after the Von Meitner Institute closed down a number of years back?
Thank you
Is
for
it being
the question.
relocated somewhere else?
I didn't want to put some introductory slide because I thought, okay, it will take a lot of time to tell the story. So thank you for the question. Yes, we stopped or shut down our reactor just 2.5 years ago, or almost 3 years ago, and so we should think for some future activity. I mean, in order to use our knowledge, expertise, and also equipment. Therefore, we we decided to cooperate with other neutron imaging groups over all the world. And there was a very fortunate need for support for the colleagues at ILL who were just going to set up a new neutron instrument at ILL in Grenoble. So this was win-win situation for us and for them. Just they could just use our expertise and knowledge and also our equipment. And therefore, we used some model of research, joint research unit, where 3 partners were put together for for the aim of constructing and building this new instrument, new imaging instrument at ILL. So the partners are HZB, ILL Grenoble, and University of Grenoble. And with this, we were able to export all our expertise to this group and to this facility, and also they will reuse parts of our instrument, which was here in Berlin. So with this, we have the possibility to continue our activity, just being member of this unit, and also to give a chance to all our users and partners which were involved at the neutron imaging instrument in Berlin to continue with their research and use neutrons again for their projects and exciting experiments.
Do we have any other questions? Okay, let's thank our speaker.
Thank you very much.
Okay, we have a follow-up question. From
a follow-up question. What is the specific beam requirements for this application?
Monochromatic neutrons.
Thermal, cold?
They can be also thermal. The problem is how efficient are your analyzer and polarizer in this case. We used solid state bender. So this means these are just lamella which are coated with super mirrors, and therefore cold neutrons are preferential. I mean, you have much better efficiency for the, let's say, for the reflection from the super mirrors for cold neutrons. That's why we had a cutoff of this solid state bender at around 2 angstrom. So this means below 2 angstrom, there will be no more efficient. But this bender can be prepared also for thermal neutrons, and this method can be used also with thermal neutrons. But in our case, we used cold neutrons because we had also cold neutrons here in Berlin, and our facility was placed on a curved neutron guide facing the cold source. So cold neutrons are much easier to, let's say, to polarize and to analyze, but with thermal neutrons, it should be also possible.
Thank you so much.
Thank you.
We've come to the end of our sessions. I want to invite Prof. Andrew to give us a summary and as announcements. Thank you.
Quite a summary. Yeah, there's no way I can summarize all these talks. I think they're way above my capability. I think we had very interesting talks. They covered many different topics, showing the diversity of neutrons. And I think that's actually what we want to show by having these talks that cover different topics within applications. Yeah, so I really want to thank all the international speakers. I don't think that, well, if they're online, very much thank you for your efforts and your preparation of your talks, which were excellently delivered. Are there any questions or points for discussion that we have? The expert is there. Jitesh can answer the question. Okay, so you're on. Is there a microphone? Another microphone? I asked this one. Let's see. Should be on you.
Just a question, in the TPR, how many of these techniques will be available, or do you envisage to have available?
Well, we're dreaming big, Johan. So yeah, I can just say this Professor Johan de Villiers from Takkies, for people who don't know him, he's from the metallurgy department, metallurgical engineering. Yeah, so we're dreaming big. So at present, as part of our consideration, are 22 instruments. But okay, that's never going to happen. So therefore, it's important to have a priority list, and that's part of this gathering, is that we can determine which are the predominant instruments that we should have. I would guess, no, I don't know who the people with the purse, or Matabo and Yanda, I don't know who's got the money, or Jitesh, or Professor Dr. Adams. I would say we should start small, like they're doing at ANSTO, possibly 5 to 10 instruments for the first round. And then of the future, of course, ANSTO did this over 15 years. Grenoble has done that over 50 years. That's why they have 40 instruments. So it should be a phased and gradual approach. So we need to identify the top instruments which there's a big demand in this country. So we can justify from a scientific case what that is. Now, it can be quite obvious. I mean, all of us are doing XRD, or many people are doing XRD work. So the first is to possibly duplicate what the XRD guys are doing. Powder diffraction, stress analysis, imaging, small angle scattering, things like that. But I think the list needs to be dictated by the community. We can't make that call. And also, it's much more difficult than that. What are the requirements for the instruments? What should their technical specifications be? And that's also dependent by the user community. The community has to define resolution, sample environments, all of these things are also important. So as Jitesh said this morning, the facility will be equipped with a cold source. That's not debatable. That's part of the instrument. So therefore, we will have thermal neutrons and cold neutrons will be part of the MPR. Of course, other sources that are also being considered are hot neutrons, specifically for the PDF, your nanocrystalline materials, PDF type of analyses. And then also fast neutrons, radiation damage in materials, gamma beams, radiation damage. And okay, Jan Reiners at the back. So we're also looking at specifically medical applications where you can use gamma beams and fast neutron beams. But it's up to the community. The community needs to become involved and be prescriptive with good scientific proposals.
And I presume that one can also use this facility sort of precursive analysis before going on to polarized neutrons or whatever.
Yeah, it's a gradual approach. So normally X-ray techniques, then neutron techniques, and then the specialized like polarized neutrons, and then spin echo. I mean, that's the ultimate technique to use. I can't see us building a spin echo. That's extremely complicated instrument. But of course, in 15, 20 years time, it can happen. There may be big demand for that. And of course, so I said this morning, the MPR is our big dream on the horizon, but Safari is in operation for the next 8 to 10 years still, and it will possibly run in parallel with the new MPR. There will be a period of sharing responsibilities. We need to come up with a program to justify the expansion of the beamline facilities at Safari 1 as well. So to present, we've got 2 instruments, the powder diffraction and the stress, and then the imaging instrument, which is under development. So I think we should make the assumption that those 3, what would the major applications be in the next 8 to 10 years? And try and fully utilize those instruments. Build our community, build our unit community, establish the expertise. I mean, Dave mentioned this morning, technical expertise. We could start building and establishing that capabilities as well, because to build this MPR, the beamline facilities, you can't buy these things off the shelf. You have to build most of the components yourself. And for that, you need exceptional technical skills. Very good mechanical engineers, technicians, software engineers, electronic engineers. All of these things are very important.
Another aspect is, we have a very strong mining industry here, metallurgical industry. And for that, one needs high temperature facilities, and more than one, which can simulate and evaluate high temperature processes, which are so important for our economy.
Yes. Yeah, and that's, Johan, I will be reliant on your network of people. We are not making any inroads in the mining industry. We're not attracting a lot of interest, and they should be. I mean, it's a major activity in this country. And possibly the post-processing after mining, adding value to whatever. So that's something that we need to consider. But we, as I say, we have not yet identified the right people for that, to assist us in that regard. A very positive aspect of this meeting today and tomorrow is the interest from the geology departments. I mean, I'm so impressed with so many people from geology, and unfortunately there's none left. That is really welcoming. We've been trying a long time to get involved with the geology groups as well, because even magnetic systems, many also rocks are magnetic. So I can see a huge application for that as well.
Thanks. My mask. I just wanted to add to that to say many of the European facilities, okay, besides Australia, the European facilities are geared towards next level processes. They're not focusing on minerals and extraction and all that because they don't have mines. This is not their core business. Their business is to add value in end products and also cutting edge research. So there's really a niche area for us in South Africa to develop that capability to use neutrons in mining and processing and all of these applications. Yeah, so I think we need to get into the mining and the geology sections more avidly and try to penetrate them. And then there's the rest of Africa as well, which we spoke about, getting African researchers from Africa to come to South Africa. And one thing to bear in mind is that if you apply for beam time at ILL or at MLZ or at ANSTO, if you've not done a neutron scattering experiment before, you can forget about getting beam time because it's very competitive. So having our facilities here is crucial and essential, as firstly to get the experience, and secondly to do the research to be able to take your proposal, etc., to some of the top end facilities like ESS will be, etc., because we're not going to get access to those places just by being at home and watching things happen. So we'll be left behind in the development space to a large degree. Thanks.
Yeah, just want to add to the comment about mining, it's not only the ores. I mean, we have some of the deepest mining activities in the world. Those cables, just think under the enormous stresses those cables are. So a lot of mechanical optimization studies can also be done, stress analyses and things like that in various aspects of the mining activities. So we should not only think about the ores, but of course that's very important because it's a big market.
I have a quick comment. You would have seen as part of the registration process, you were asked to complete a whole list of questions. So on that was which instruments you would use if it was available. So I've reopened registration process. So during this course of the 2 days, if you see there's something you can use, please go and change your options or add to them, because that will also be an indication of how well this meeting, what change this meeting has gone. So please go and revisit your registration as well. Thank you.
Yes, that will assist us in this priority order that we need to determine. So I'm sure people, the reason for these talks, the way they were structured, is to give a background of the applications. And in that, the user community can identify fields of interest. I mean, we don't know what those fields of interest are. So based on this expert talks, it's now easier to make this link. And therefore, please reconsider your assessment of instruments or techniques. And I'd say even keep it open until lunchtime tomorrow when all the talks have been completed, because there are very interesting talks tomorrow as well. And then reconsider doing that assessment, please. Yes, Dave.
Hi, Andrew.
You're starting up so many thoughts in my head at the moment about how to build a user community. And look, I've been trying for decades now how to nurture higher end powder diffraction. Johan and I have walked a long path on it. There's a small group of us that do it. And I think it would be great to couple that with neutrons and sort of beneficiate. So it's not just the neutrons, it's the synergy that we can build a single larger community that goes after these kinds of structure property relationships, which is largely what we are looking at. Then on the thought about so once we've decided that it will be a cold thermal neutron source predominantly, I think we need to look at how we build the niches off that. I don't think by default, yes, I am partial to wanting to do PDF studies and stuff, because that's part of where my research takes me, but it is to optimize how we can go after it. So if it is good for us to build, say, a high resolution, fast powder diffractometer, I think that could add far more to a community than to go after a niche more complicated like PDF. So keep in mind, it's not just, there's many factors in an instrument that one has to consider that we can go. So I would probably say I'd like a fast, higher resolution powder diffractometer more local. And that would bootstrap us into many things. Bringing in mind, I then want variable temperature, like Johan said, and to do the magnetic stuff, you want to do it from probably like 3 Kelvin.
Millikelvin, actually.
Millikelvin, yes. Right up to 2,500 Kelvin. It's, yeah, that I think would make a lot of people happy.
Yeah, thank you for the comments, Dave. So I know that you are a predominantly X-ray and a synchrotron person, but these techniques are not in competition. So we need to work together as a community. I mean, it's the applications that become important, using different radiation types in our investigations.
Sorry, I was just saying it. These probes it's like you have your neutrons, your X-rays and electrons. They have pros and cons along the line, each one of them. And if you really want the full picture, you want to see all of them, and you want different probe sizes and everything out of the whole lot. And then, of course, you want to do a lot of imaging. I would love to see more imaging. I think it's not something I'm specialized in, but for me, that's a slam dunk application. It's there for everybody.
Chachi's hand up.
Thanks, Andrew. Actually, it's Mark online.
Mark Johnson.
Yes.
Okay.
Mark,
I'm pleased to know Mark's still with us.
Thank you. No, it's a really, really interesting and exciting discussion, because you're looking forwards with ambition. So I think that's great. There's a few comments that I'd just like to echo, really. I think the first one was that, you know, you focused on the sort of the need for expertise that you've got to develop, not only to build the instruments and develop the techniques, and those techniques specific to South Africa, but also develop the user community. And I'm sure it's true for all of us who've been talking today, but certainly I can really only talk for myself at ILL. You know, we're ready and very keen to help you on that path. And as somebody just said, you know, at ILL, it's been going on for 50 years, and we've got an instrument suite of 40 state-of-the-art instruments. And that gives you a very good view of what's possible and you know, and what can be transferred to you and developed quickly for you, and then what's maybe specific that you would like to bring to the neutron, the broader neutron community. So the first message is, you know, we're very keen to engage with you and help you, and you know, and share our expertise. And the second message I think is really important. It's these more recent points about the sort of the broader view that you need neutrons, X-rays, electrons, and so on. I've been in the process over the last year of engaging with our scientific members at the ILL in various ways, various meetings. And what strikes me with those various countries, and there's 14 of them in total, is that many of them do bring together the neutron and X-ray community. You know, they're interested in scattering techniques. And while neutrons tend to be the smaller partner, simply because, you know, there's no lab-based neutron sources and there are fewer neutron sources than synchrotrons, I still think it's a very good way of proceeding. And, you know, you bring together a broader community. And I think we should always aim to attract people from the X-ray community into the neutron community, and make sure our capability is being fully exploited in that way. So I think that's the second main message from me to you, is that, I mean, you think just neutrons in terms of a facility and the instruments and the science that you might do at that facility, but think more widely and engage with your X-ray community if one is well formed already, and sort of work together with them on developing the use of large scale facilities.
Thank you very much, Mark. Any comments to what Mark has tabled, Robert?
Thank you. I've got a question for Mark, actually. Mark, you presented this morning about in your presentation, you mentioned there's now a drive for use of neutron imaging trying to replace SA-SENS. Do you know the drive behind this change? Thank you.
I don't see it as neutron imaging replacing SANS. They're very much complementary. I mean, SANS sort of covers the length scales from nanometers out to microns, and then imaging, which at its best resolution starts at a micron, and then you image in real space directly, as Nikolai's shown. It's always been a mystery to me why ILL, we took so long to get into imaging, but we weren't the only ones who were slow on the uptake. You know, ISIS has recently developed their imaging activity, and it's just been said it's a slam dunk technique. We're actually already planning to build a second instrument to complement. The first one is massively oversubscribed both by academia and industry, and I think we're aiming to build a second one quickly, and, you know, I could imagine a third one would be possible. So imaging won't replace SANS. SANS is a hugely important technique. Throughput is high, productivity is very high. Our SANS instruments, the three of them, produce more than 100 papers a year. And of course, a large fraction of those are of the highest quality. And imaging is just growing substantially and very rapidly. And I think we've got to try and ensure that we have sufficient capability. And so it's great to know that you've got an imaging project underway at Safari 1. I think that's really the right thing to do. And it's one of the key techniques, along with SANS and powder diffraction, to start with, I think, at your new facility.
As follow-up questions, you just showed a little bit of a income. Is it the drive behind? Is it the income to drive behind to build the third instrument, or it's just now industry is coming to the board to use utilizing the instrument? Because you showed about €1 million per year.
Yeah, that's what our potential is at the moment, based on the recent experience and sort of provisional orders for beam time. You know, we're primarily serving the academic research community, and so there's not a direct connection between industry revenue and building new instruments. The academic use of the imaging instrument NEXT, it's already oversubscribed by a factor of 4 or 5. So there's a huge amount of excellent science to do, and we should, independently of financial considerations, build another imaging instrument as quickly as possible. And that's what we're trying to do. And but of course, having said that, we all need money to operate and deliver on our full potential, and the industry income will certainly help in that context.
Thank you.
So nowadays the instruments are very close, as is there. So nowadays the instruments are very close to 1 minute or 30 seconds per diffraction pattern. So they are moving into this dynamic range. So high intensity diffraction, neutron diffraction really exists, but you need a very fancy detector for that, of course. Okay, so I just want to then close this session. Hopefully our geologists are still in the audience. So there's a talk tomorrow on geological applications. And if you look at the program, you'll see there's no title in that. So I just want to tell you what the title is. So it's Neutrons and Geoscience. It's innovative research and scopes for understanding Earth's systems and materials. Sounds very impressive and interesting. So yeah, thank you very much for all your participation today. Please keep that energy for tomorrow as well, and enjoy your evenings, and we'll see you tomorrow. So we start tomorrow, I think, at 8:55. The cocktail function, it's down the corridor. The first, where the toilets are, that corridor there, passage, it's down there, and you walk into the venue. And you can, of course, with a beer in hand, have a view over the lake and mesmerize and whatever. Thank you very much. Thanks for your participation.